Hello, everyone. Welcome to our Foundations of EEG workshop for 2025. We have an exciting program lined up for you today. Today on the theory, hardware, and application principles of EEG. Today we have people joining us from across the world. So to give you an idea of where we are, our Brain Products office is in South Germany. More specifically, we are located in the town of Gilching, just outside of Munich. And we're happy that we can reach you all from here. My name is Shiva. I'm in the team that takes care of Brain Products Academy, and I will be your moderator today. So I have the pleasure of introducing the speakers for you who will be speaking to you today, who you can also see on the screen. They are members from different teams at Brain Products and are very knowledgeable about different aspects of EEG hardware and software. Most importantly, they're all scientists with a research background. So they are all here to help you apply EEG effectively to your research. Let me introduce you to them. David leads our technical support team. He worked at multiple labs evaluating the reliability and impact of different experimental methodologies. Michael is in the scientific support team. He trained in psychology and his research specialized in the cognitive neuroscience of learning and memory. Lena is also in our technical support team. Her training is in neurobiology and behavioral physiology, and she investigated impulse control in different psychiatric disorders. Fernando from the scientific support team has a background in psychology and neuroscience, and his research was on the visual motor interactions in different clinical and sports settings. And Laura from the sales team has a background in clinical psychology and systemic neuroscience, where she combined EEG with different methods like fMRI and eye tracking. So thank you to our speakers for being here today. So what will we learning? What will we learn today? Hopefully by the end of the day, you will be able to answer several questions. What is EEG? What do you need to record EEG? How to choose your amplifier and electrode technology for your research, and what software do you need for recording and analyzing EEG. For those of us who will be joining us for the full workshop, we will be going into more depth over the next couple of days into different topics: filters, pre-processing, triggers, pilot testing, and experiment design. And in case you aren't unable to join us then, you can always sign up for the event's mailing list. You can scan this QR code here and to receive updates on upcoming workshops and webinars. And in fact, I can tell you about a few that are lined up already. We have 3 workshops focusing on hands-on experience. The first is in Hamburg from May 20th to 22nd that covers a broad range of topics from basic to advanced topics in data analysis. And then another workshop from June 11th to 12th in Munich focused on hands-on experience in EEG data acquisition. And this workshop will also be conducted in Australia if that is a more convenient location for you to join us. And you can scan this QR code and learn more on about these workshops and how to register. And at this time, it's time for me to stop talking and essentially hand over to David and Fernando, who will be telling us about what is EEG. And David will be the first speaker who will be taking over.
Thanks, Shiva, for the nice introduction. And me get my setup ready. So first, also for me, hello everyone. It's nice to see so many of you like digitally in the attendee list, and I'm looking forward to hearing your questions or reading your questions later in the first Q&A, and also seeing you later in the breakout rooms Shiva mentioned. And as at least this first talk, or the first talks, will also be later uploaded to YouTube. Also, hello YouTube if you're looking at a recorded version of this talk. So I will be talking about what is EEG, so the thing we are all interested in here. And the short answer is, of course, EEG is an abbreviation, not for the German Erneuerbare Energiengesetz, which you might sometimes read, or it's not EEG. It's an abbreviation for electroencephalography, which is a bit of a mouthful, so I will stick to EEG most of the time. But to briefly explain this electroencephalography term, it's consisted of three separate parts. The first is electro, it's basically the electrical activity. Enkephalos from the Greek means in the head, so in this case the brain. And graphy, also in the original Greek sense, means writing, but in our more abstract way, it's also now a recording. So a recording of the electrical brain activity is what we're interested in. And why are we interested in it? So the basic question EEG tries to answer is, how do we get from here, so some sort of brain reading, to here, which is basically a deeper understanding of the mechanisms of our brain. I think I still hear Shiva in the background, if you want to mute yourself. Yeah, so how do we get from the brain reading to the understanding? Via recording of these squiggly lines from the top surface of the head. And the first thing to answer here, or to look into, is how this recording even takes place and what are we recording here. So a bit about the neuronal background. So if we zoom into not just one neuron, but basically the first building block is just a synapse, so where two neurons meet, because the communication between neurons is mediated here. If an action potential arrives at the presynaptic terminal, it leads to the release of neurotransmitters in the synaptic cleft that then diffuse through this cleft until they reach the postsynaptic membrane, where they trigger a postsynaptic excitatory potential, which is the main source of the EEG. So it's not mainly the action potentials, but these excitatory postsynaptic potentials, which is also a mouthful, which is why I usually abbreviate them with EPSPs. Because these EPSPs then generate dipoles at the apical dendrites. So these are the apical dendrites of one neuron, where the EPSPs are generated. And yeah, but also we don't only look at one, but we're looking at multiple of these cells. So if we zoom out to a little bit at least, to the neocortex. Neocortex is organized in several layers, in this case 6 layers, with different characteristics. And the majority of the EEG signals are generated by these pyramidal neurons, which are very neatly and parallel organized in these layers, primarily in layers 3 to 5. Because of this parallel organization of the neurons, the signals from all the different neurons sum up more or less nicely at the top, so we can record an EEG signal of summed up activity from multiple parallel neurons. But before we reach this layer, we have to zoom out a little bit more. To reach the scalp electrodes, it has to cross several layers of non-neural tissue with different conduction properties that also attenuate and might change the signal in various ways. So first, there's a cerebrospinal fluid surrounding our brain, the different meninges, so all these maters here, arachnoid mater, dura mater, pia mater, then the scalp, and so on, until we have then the electrode, often also with first a layer of electrode gel and then the electrode on the top of the head. So, and finally, if you zoom out even more, we usually don't only have one electrode, but a set of electrodes located around the various positions on the skull that then record the brain activity from several, or a lot of these pyramidal neurons distributed around the top layer of the brain. And what do we measure here? So the one metaphor you can think about is we are measuring a synchronized firing of big groups of neurons, which is a bit like observing a sports stadium from above. So if you would imagine hovering about a sports stadium, you don't hear one single person talking or clapping or doing anything. You only hear if a large group of people react in a similar way, maybe a goal is scored or something significant happens in the stadium, and at least half of the stadium is usually happy about this. Maybe the other half is not, but at least a large part of the attendees here would maybe break out in synchronized clapping, which you can then hear or maybe even see if you're hovering above, but you cannot make out a single person. And similarly, you cannot make out a single electrode with at least surface level EEG. And in this case of the stadium, why do they clap? It's because something happens, or in the case of neurons, why does a group of neurons fire together? There are also two ways of thinking about this. One would be a summed up evoked response, which is basically similar to silence, so nothing happening, no one's clapping. Then something happens and everybody starts clapping at once. But you can also think about phase resetting of ongoing oscillations. So maybe people are already engaged and already clap or make noise, but not synchronized, which you also cannot really make out because it's smoothed out over the whole stadium or the whole brain. But then something happens that brings them in line and they all start clapping at the same time, and this is much more noticeable, which is a way of thinking about phase resetting. So if we now have this group of neurons firing, how is this represented on the surface level? So at the very basic level, we can think about just one group of neurons generating a dipole, which is a way of symbolizing the electrical activity. And I've visualized here it in different ways. First, on the left side, you see the brain from the left, the right, the top, the bottom, the front, and the back. And you see one idealized group of neurons making a dipole here, which is always the same, perpendicular to the surface and right at the center of the brain. And then you can look at the time course and the topography from the top of the brain. It's also often you see a very minimal brain, so you only have a nose to see where the front is and the ears to see where left and right is. But that's basically our top of the head. And here in the middle, you see the dipole, and then the topography with positive deflections all around this dipole, that it gets lower and lower the farther away you go until it switches sides and is negative on the very outer shell. You see also the time course here. And this would be an example recording from various electrodes. So if you have a central electrode CZ right above the dipole, that of course records the largest activity. The farther away you go, the less pronounced the positive activity is, and if you are even farther away, you see even a negative activity. But of course, we only have one dipole here at the very center, where it's not very sensible oftentimes that you have this activity exactly here. So what happens if this activity comes from a different point and maybe even a different orientation? And that's what I simulate here in a short video. You can see if I tilt the orientation, not changing the position, you see the topography changes, and then I change the location as well, and you see again resulting topography change. Tilting the orientation again also tilts the topography. And with this example, you have one more or less auditory signal here in the on the right side. Also not perfectly perpendicular to the surface, so you see the topography is shifted to the front, even though the source is relatively in the middle. And also we usually don't have only one example dipole, but we have multiple. So in this case, let's say we have an auditory experiment and people hear the same on the left and the right. So we would have at least two dipoles, one on the right, one on the left. Maybe not exactly at the same position because brains are different. And also maybe not with exactly the same time course. So the time course on the right is a bit sharper, on the left it's a bit more attenuated. So the resulting waveform would then be the superimposed of these two dipoles. So you have the left and the right superimposed on your EEG. If you go one step further and add another set of dipoles, maybe this time in a more or less visual potential, which arrives in the posterior region and then travels a little bit further to the front. I simulated 4 dipoles here, left, right, and then a bit more central. Also, the first set of dipoles has a positive deflection, and the second set of dipoles has a negative deflection, and all these 4 then get added up, which slowly begins to look like a real EEG. If you already have seen a real EEG, where you have a strong activity in the back here and a lower activity in the front. Also a positive and negative deflection, which is similar across left and right hemispheres, but not exactly the same. But one additional thing to keep in mind here is for one, all these sources are similar, but not exactly the same, and they overlap both in time and in place. So all these 4 topographies, all these 4 time courses overlap. And of course, we don't only have 4 dipoles active. The brain does a lot of things, not only reacting to one stimulus, but we usually do more than just that. And one way of visualizing this is usually noise, which is basically just saying there's a lot of other signals we are not interested in and which occlude our signal of interest. So I just add some random noise here, and now it looks even more like a real EEG. Of course, this is just a simulation, but to simulate the other way around, if we think about these 4 sources and build up the resulting waveform. Usually we do the other way around. We have these waveforms and think about where they come from. One step further is also this orientation I wanted to highlight here, because the brain is hopefully not smooth, so there are some disorders where it is, but usually you have basically valleys and mountains, or in the case of the brain, we call them gyri and sulci, one gyrus and one sulcus. And if you have the activity on the mountain or the gyrus, it's very straightforward. So you have a perpendicular source here. The topography is neatly organized on top of the source, and you can relatively easily make the relationship between the two. If you have a tangential current, it gets more difficult because the source has to travel a lot of space before it reaches the surface of the brain. So the topography is, for one, shifted, and for another, it's also weaker. And the oblique current is basically in between the two. So one fundamental challenge of the EEG is that you have a very, very tiny signal. So you're looking at activity of neurons that have like in the microvolt range. It's also overlaid by multiple other signals. It's distorted by various influences, and it's variable across subjects and trials. And there are various solutions we will go over today. How to deal with that. One would be to use appropriate EEG hardware, which I will talk later. You can filter it. You can employ good study practices in piloting, or pre-processing strategies and artifact removal. But one main solution I want to talk about briefly here is averaging across trials and subjects. And what does this mean, or how do we get there? It's basically one of the most basic paradigms that you show 2 different stimuli, let's say stimulus A and B. Maybe one stimulus is emotional, the other is not, and you want to see the emotional difference or something else. But you don't only show one stimulus to one participant one time, but basically multiple trials on one participant and multiple participants, and repeat the presentation multiple times, and then average it together to then look at the difference in the average waves to meaningfully interpret, okay, these 2 signals are somehow different, and this tells me something about how the brain perceives them differently. Because if you only look at one trial from one sensor, you might not see much. So this would be one example trial. On the x-axis here, you have the time. The y-axis, you have the voltage. And this is basically the EEG signal. And you don't have a clear picture from one trial alone. And if you look at multiple trials, they might look very differently and almost completely different, but not totally completely different. For example, in all 3, you might see this negative deflection here around 350 milliseconds or so, but it's different every time. So you need multiple trials to increase your signal to noise ratio, with the reasoning being that artifacts or noise is not correlated to your stimulation. The brain, all the other things the brain does, and also the environment around your EEG does, do not care about the stimulation. But your signal of interest will be similar each time you present the stimulus. So it will average out the artifacts and increase the signal to noise ratio of the stimulus until the average shows the stimulus more clearly. So this would be one average waveform. You can also, again, look at the waveform in different ways. One would be, again, just looking at the eventuate potential. But oftentimes we also average the one time window of interest where we see a strong effect and show the topography again. So again, the brain from the top, and show in colors if you have negative in blue or positive deflections. These are just 2 examples potentials you might see, the early posterior negativity. So the name implies it's early, 200 milliseconds. It's posterior, which is in the back of the head, and it's negative, which is blue. And the late positive potential, again, it's later, 400 milliseconds. It's positive, which means it's red. And it's more central also. But if you only look at these averages, you might miss all the background. So where does it come from? So I want to zoom out a little bit as well. So first, just looking at this group effect over time tells you a bit more about how these effects come to be and then vanish again, because it's not a clear process that they suddenly appear and then disappear. But you can see that the EPN already builds up a little bit before the main effect and then also stays a little bit after the main effect, similar to the LBP that slowly gets stronger and weaker again, even though you might only zoom into one picture here. Also, to briefly show the variability across participants, I plotted just the first 11 here, and you can see again the EPN looking different in each participant, similar but different. For example, for participant 6 here, the EPN might be a bit later, while, for example, for participant 10 here, it might even be a bit earlier. Also, the topography looks different across participants and so on. So a single participant does not always show a representative signal. The orientation of sources differs between participants, so the topography differs. So you need multiple participants for general conclusions. Last thing I wanted to talk about is just to visualize the order of magnitude we're dealing here. So the exact numbers here might differ, but I just plotted one example here. Let's imagine you run a study with 128 EEG sensors. You look at 1,000 milliseconds of time, or sampled at 1,000 hertz, or 1,000 sample points. You repeat the simulation with 100 times. You have maybe 32 subjects because you want to split half female, half male, half reacting left, half reacting right, and so on. And you have two conditions you're interested in. And if you add all these up, or rather multiply all these, we arrive at around 1 billion data points. So the exact numbers might differ. You might have 10 times more or 10 times less, depending on the exact numbers you have in here. But just to give you a sense of scale of what we're dealing here. So a lot of people talk about big data, and in a sense, we in the EEG world can also talk about big data. We have a lot of data points. So one fundamental question is also how to visualize all these humongous amounts of data. Of course, these are not independent of each other. The sensors next to each other are very different. The time points next to each other are very different, but still there's some degree of variability between each of these data points. So it's very important to visualize them in a clear and distinct way without forgetting what the huge amount of data that's behind them. And I just want to briefly allude to 4 options here. Fernando will go into more details later. So the most common EEG measure is the time domain measure, which I also showed earlier. For example, the event-related potential basically averages the data in one or more sensors over time, and you see this waveform. But you might also transform the data in several ways. For example, with a fast Fourier transform, or FFT, you might look at the frequency spectrum. So not having the raw data, but rather the 5 hertz, 10 hertz, 20 hertz data. Usually you have a deflection in 10 hertz, which is one of the most prominent brain frequencies. Or even more complex, looking at not only frequency or not only time, but rather the time frequency domain. For example, this event-related desynchronization, where you have a more complex way of looking at it. And even more complex might be higher order and non-linear measures, for example, looking at connectivity, that one brain region precedes and drives another brain region activity. But I won't go into too much detail here, but rather give over to Fernando, who is our expert in data analysis. So he will tell you more about what EEG actually is and how it looks.
Yes, so thank you, David, for your introduction. So I would now like to continue with our presentation of what is EEG. And for this, I would like to place EEG in comparison with other techniques in neuroscience. So here we can compare the different characteristics of different neuroscience techniques in terms of their spatial resolution and their temporal resolution. So here we find first the EEG and the brother of EEG, the magnetoencephalogram, or MEG, in terms of having very high temporal resolution, so on the basis of milliseconds or even faster, but relatively low spatial resolution, which means that we cannot tell exactly where in the brain an event that we observe is happening. If we bring this in comparison with the techniques of FNIRS, of functional near-infrared spectroscopy, or the fMRI, or functional magnetic resonance imaging, they have a higher spatial resolution, but a lower temporal resolution. And here we have other techniques which are invasive, the ECOG, the electrocorticogram, and the LFPs, which are recordings that are performed intracranially, but they are invasive. They have very high spatial resolution and also temporal resolution. However, well, as mentioned already, they are invasive and they have to be usually performed in patients that already have this kind of electrodes implanted. And the recording is also restricted to the location where we have the electrodes implanted. So seeing it from other perspective, we can compare more of the characteristics of the different techniques over here. So here we have first the EEG, which measures the electrical activity, having a high temporal resolution, low spatial resolution, and having a relatively affordable price. And also, it's a technique that can be quite portable with the equipment. There are different kinds of equipment, but overall, they can be brought to different places more easily. While in comparison with the fMRI, now we are measuring the hemodynamic response of the brain, and the temporal resolution is low because of the technical implementation and also because of the time course of the hemodynamic response. But as mentioned before, the spatial resolution is higher, but of course we need a much larger installation for this kind of equipment, so the price is more expensive, and it's of course not portable because it needs to be installed. And if we bring now the FNIRS, which is also measuring our hemodynamic response, it's somewhere in between the previous two. So it has a relatively high temporal resolution with also relatively high spatial resolution, but not as high as the fMRI. And it's also more restricted to the cortical measurements. And the price is somewhere in between in the range. And it's also more portable in the mobility. So in this case, we can see how these different techniques complement each other and can bring different aspects of the brain activity, and also have pros and cons, each one of them. But if we overall in research use them together, we can bring more and more of a complete picture of a brain phenomenon that we are investigating. And of course, nowadays we have the possibility of multimodal imaging, where we can combine more than one of these at the same time to gain more information about the phenomenon that we are investigating. So overall, this gives us a perspective of where EEG is standing together with other techniques in neuroscience. But now going more deeply into EEG itself, well, it's electrical measurement of brain activity, but from here we can extract different kinds of information as well. So depending on our research question, we can obtain, for example, information about localization of where in the brain an event is happening, the timing of that information. We can also extract frequency or the brain waves of the event that we are observing, and also investigate interchannel dependencies, or how different regions of the cortex, in this case measured from the scalp sensors, are interacting. So which one of these measurements should we use? That depends on what do we want to know or what we want to analyze. So do we want brain activity that is specifically located around specific events in time? Or do we want to investigate more long-standing states of the brain, transient and frequency-specific effects, or time-locked? do we want to know the origin of the signals that we are investigating or estimate their source in the brain? And do we want to know the interplay of electrode or source reconstructed signals or analyze them independently? From here, we also have an effect on what kind of experimental design that we will use in order to enable extracting these kinds of information. So in this case, we are seeing the two different kinds of paradigms that we can use for recording the EEG, which are the event-related, based on trials, or block design for states based on longer periods of time. Then to further refine the information that we want to extract, we can analyze this information on electrode space or on source space. And further, the specific signal that we extract can be the ERP, event-related potential, frequency using FFT, time frequency using, for example, wavelet analysis, source localization using, for example, the Loretta analysis, or connectivity analysis for interactions between different regions using coherence and correlation kinds of analysis. Let's take a deeper look into the kinds of designs that we can use. Starting with the event-related paradigms or event-related designs that we just mentioned. In this case, we have our stream of EEG recording, and along the EEG recording, we have different presentations of a stimulus, like stimulus A or stimulus B, and some of them may require a response from the participant, but of course, it can also be that there is no response. And what we're going to analyze is the reaction that happens around each one of these stimulus presentations or response, each one of them considered as an event. And then we will bring them, average them all together in order to more clearly see the kind of brain response that we're interested in. But what is very important in this design is that we have a very specific marker in time and very precise for each kind of the events. As we already saw with David, we need a larger trial number in order to be able to average them together later. And also we need to consider the duration of each trial or how much activity around the marker are we going to analyze so that we have enough time to let the brain generate a response within each one of the trials. It's also very important that the presentation of these trials is randomized, because if we present, for example, all of stimulus A first and all of stimulus B second, then there may be the case that we have habituation effects, and this affects the brain response to each one of the events. Here, second, we have the block design paradigms, in which rather than having specific markers, we are measuring over longer periods of time. And in this case, the entire stream is what we're interested in. We can have different conditions here, like, for example, eyes closed, eyes open, concentrating, not concentrating, or before medication, after medication. And we are interested in the running activity all along. Here, what is important is that we have start and end markers so that we know when we have each one of the blocks. And we also need to consider how long is the duration of each one of the blocks to gather consistent activity, and how many blocks. Is it only one block of each one, or do we need to repeat them in order to have stronger effects? As before, it's also important to randomize, even though they are longer blocks, we need to randomize across subjects which one of the blocks goes first in order to have a better experimental control. Now, after these paradigms and kind of designs, let's get into the kind of signals that we can extract, starting with the event-related potential, of which we have already seen some examples. But to check more clearly why we need to have a repetition of these events many times, that is because with each one of the trials, we get together noise, which we have here in isolation, and our signal of interest, the ERP, that is also here in isolation. But with each one of the trials, we get them both together. But if you notice, this signal is included in here, but because of the noise, we cannot see it very clearly. So what we need to do is to have many trials so that this evoked activity, or the one that is produced by the event, when we make an average, it will persist because it's more or less consistent. It may have a little bit of variation, but it will be consistent enough so that if we make an average, it will persist in the average. However, the noise, because it's random, then it largely gets nullified. That means that when we have both of them together and we make the average of all the trials, then we can see this ERP, or event-related potential, more clearly, while the noise is largely nullified. So why would we do an event-related potential? This is when the effects that we want to analyze are very restricted in time, and they are revealed to the averaging across the event, like the stimulus presentation or the response that the participant is doing. And we can use this to compare conditions, like, for example, under pressure, without pressure, or patient group, control group, and find differences in these ERP components. What they represent is local synchronized neural activity over time related to the event, which, as we've seen, is revealed by averaging across the trials. From here, we can analyze different parameters like the amplitude of the ERP component, the latency of the component as well, and also the distribution of the component across the scalp electrodes. Like, is it, for example, here, the P1 stronger over this region? And as time evolves and the different components of the ERP appear, then we can see them, the strength of them vary across the scalp. So I would like to talk about the nomenclature, or how we give names to these ERP components. And the first thing I would like you to notice here is that we have the polarity, where positive is down and negative is up. This is has been a tradition in ERP and EEG research for a while, but you may see it the other way around as well, like positive up, more traditional of science. So whenever you see an ERP plot, it's important that you check where the polarity is to interpret the signals. Then with the different components, one of the most traditional ways to name them is by their order of appearance. Like in here, we have P1, meaning the first positive, N1, first negative, P2, N2, and so on. You may also find them as P100, N100, and this is based around the timing when these components appear. But the timing may vary a little bit, and that's why some researchers prefer to name in this way P1, N1, and so on. In one ERP waveform, then we get the different components, of which I would like to show you different examples, starting with the very early components that are so small that we cannot see them here, where in the case of a visual stimulus, we get at first the C1 potential that changes polarity depending on where the stimulus in the visual field is presented. Or in the case of auditory stimulation, we get a series of deflections in the auditory brainstem response. But these ones are very small and need a very high resolution, so we do not see them in this schematic here. But afterwards come sensory components that are still processing the stimulus as it incomes, but they are mostly affected by the physical characteristics of the stimulus, such as the loudness or the brightness, but also, for example, by the state of arousal of the participant who is perceiving them. Afterwards come the more long-lasting cognitive components that reflect the cognitive operations around the stimulus that we are perceiving. not only affected by the physical characteristics, but more of the way we decide about this stimulus, for example. And here I would like to come with one example, which is the N170, which is in a way an in-between component between sensory and component, because this component reacts to the perception of phases, which we can see here, for example, a larger deflection of this N170 component for phases in comparison to House's stimuli. So this already, it's sensory, but it's already classifying in a way that we are perceiving something that we are very specialized in perceiving, like phases. Now, moving into other ERP components that have been studied, we have the N2, which is a special case because different kinds of phenomena have been observed in this N2 time period. One of them is or also called the N2A, or mismatch negativity. It appears in the case when we have a different kind of stimulus appearing in between a normal kind of stimulus. So if we are, for example, presented a standard stimulus of 1000 Hertz, and all of a sudden, sometimes comes a deviant stimulus that is different, then we get this mismatch negativity. With greater difference with the standard stimulus, we get a greater mismatch negativity, as we can see here, for the standard and for the deviant with the discontinuous line. Then if we make the difference between these two wavelines, then we obtain or can isolate this MMN, and it's larger for larger deviants. The N2B appears when we have to withhold a response, where we have the apparition of different stimuli, and one of them is less frequent, and for this less frequent stimulus, we have to refrain from responding. So for a go stimulus, we press a button. For the no-go stimulus, which is more scarce, we have to refrain from responding. And when we have to do so, then we can see this N2B component. And it's larger, again, when we have less frequent stimuli. Similar, we have the P300 component, which appears when we have an oddball or a less frequent stimulus, but in this case, we have to respond to that less frequent stimulus. And in that case, when the stimulus appears, we can see the P300 response. But it can also appear without a response, for example, if we have the presentation of our name in between other names that are not like ours, for example. A different kind of ERP component, which is more of a slow fluctuating wave. And this one will appear, for example, in comparison to motor response. It's called the readiness potential, or in the expectancy that one stimulus will appear. So in this case, participants would make a button response, and afterwards we would have a stimulus. So we can see how there is a deflection in preparation of the button press. And then as the participants expect the stimulus, we see also this is slower deflection. The slower deflection is called the contingent negative variation, and the preparation for motor action is the readiness potential. We also have the ERN, or error-related negativity, which is special because this one appears after we execute a response. And this response is larger, or the ERN is larger, when we commit an error. There are other ERPs for linguistic or phenomena, like, for example, the N400, which appears when we expect an unexpected word. Like, for example, if we are told that there is a tropical place with palms, there's no surprise. But if we are told that there are tulips, then this is surprising, and we show this N400 component. But it can also appear if the context changes in other ways, like, for example, if we have a certain pattern of line drawings and then there is a different one all of a sudden, we also get this kind of component. Then we have the P600 component for for example, grammar errors or syntactic violations, where we have an anomalous grammar, and we show this kind of component more prominently. So we have to be careful with the interpretation of ERP components. It always depends on our experimental design and context, like, for example, how complex the task is, the characteristics of the stimulus, the instruction that we gave the participant, and other interactions like were they multitasking or having other kind of responses at the time of the ERP. They also should be interpreted across or in respect to the subject group. So it's not the same in healthy adults as in patient populations, and it also changes across the lifespan. And we also need to take in consideration the recording and analysis that we make. Like, for example, was it daytime, nighttime, or was it on the field or in the laboratory, and what kind of EEG reference that we used. But besides ERPs, then we have that, if you remember, these longer standing waves of the EEG. And the EEG can be considered as the summation of different sine waves, which are behind the noise that we observed. And this brings us to the different kinds of brainwaves that have been studied. The most famously one, the alpha that appears when we close the eyes and we can observe with our plain eyesight on the EEG, but we can also apply different kinds of analysis of these brainwaves. So they have been analyzed in different contexts, and broadly speaking, we have the different bands, like the gamma band, which has been related to high focus, beta for active states of mind, alpha for quiet resting, theta in drowsiness, and delta for deep sleeps. But there's more context than this for how these brainwaves appear. They can also be used to infer connectivity analysis between different regions, as we analyze a relation between the oscillations in two regions. And the question is how or which kind of analysis do we use to extract this kind of information? So typically we can use the Fourier transform, where if we consider the different signals that we have, and we consider the EEG as the summation of sine waves, then we need to have our summated signal divided into different time periods. And then what the FFT is going to do is to probe different sine waves to check the similarity of the sine wave with the complex signal. Whenever we get similarity between our probe sine wave and the signal, then we get a larger coefficient. So we can use it whenever the EEG is more or less stable to obtain this kind of coefficients and know if there is large degree of this band activity. So this can be, for example, when we have a concentration period, resting period, or as I said before, for before medication, after medication, but we are expecting stable oscillatory effects over time. What we obtain out of it is voltage power or voltage or power of the different frequencies or frequency bands. However, we have one disadvantage here, that we are assuming that the signal is stable. But what happens if the signal is not stable, like in this summation of signals that are modulated, it gives this complex signal. And if we try to do the FFT, we get different results at different sections of the signal. To compensate for this, we need to resort to time frequency analysis, which is able to show us how the signals are modulated over time. Like in this case, the 30 Hertz signal is modulated, and we can see here the modulation being reflected as well as for the other signals like the 8 Hertz or the 1 Hertz signal that we see over here. So we can use this kind of analysis like wavelets or complex demodulation or Hilbert transform, among others, to be able to tell how the frequencies are changing across time. and it can be done in an event-related fashion or also in a continuous fashion. We will obtain the voltage across time for the different frequencies. Now, coming into other kinds of analysis, we can also check our connectivity analysis using this same kind of oscillations. And there are different kinds of analysis, like, for example, the coherence or correlation analysis that allow us to check for possible interactions between regions of the brain, although in this case, they don't tell us about the direction of the interaction. An example is with coherence analysis, where if we get a consistent phase relation between two signals, then we will get a larger coefficient. And finally, we can also resort to the source localization, in this case with the Loretta analysis, in which basically if we know the characteristics of the tissue and the brain activity, we could estimate how the EEG would look like. But if we do this in reverse, so we have the EEG and we make some assumptions about the tissue and the conductivity of the tissue, we can estimate the source of the brain activity, for example, of an ERP. Then I would like to summarize by concluding that your research question determines what kind of activity you are going to, or what kind of analysis that you are going to use. Also, if your experimental design will be event-related or a block design. And well, very often we need more than one experiment or more than one analysis in one experiment to answer our questions. So it's a good idea to be well informed from the research about which approach we're going to use. So with this, I would like to thank you for your attention. Here is our references, and give time for some questions.
Thank you, David and Fernando, for this very nice talk. And we have some questions for you. So let me first ask, so Joseph had a question for you, David. And he wanted to know, could you explain the difference between the positive going voltage and the negative going voltage a little bit more?
Yeah, so as Fernando also told you before, I think there's also a different way of looking at the same thing, essentially. Because, for example, it's already starts as how positive and negative is even displayed. Sometimes negative is up, sometimes positive is up. I will go into a bit more detail about how we record it in my second talk, which might become clearer. But basically, what we're looking at is always a difference wave. So we're always looking at the difference between one electrode, usually the reference electrode, and the signal electrode. And it basically depends on how these are positioned around the signal we are wanting to measure. For example, if one is positive and one is negative in a certain configuration, and we switch sides, we see the reverse signal, which is still the same signal, just because we are looking at from a different set of positive and negative or subtractor positions. Also, the second point is if we have a positive deflection somewhere on the brain, we also always have a negative deflection, because our brain doesn't get charged by thinking, luckily, or maybe not luckily, but so for example, if you have a positive source somewhere in the center of the head, you will have a negative source around it, or vice versa. If you have a negative source on the front, you will have a positive source on the back. So basically, the sum of all activity is always also zero. So positive and negative are important, but they don't mean that much, because it just depends on how you look at it, basically.
Okay. Because there was a somewhat similar, but maybe not exactly the same question from Subhashini, who asked, when you show these topo maps, when you show the dipole simulation, what would the blue or red indicate in terms of a stimulus? Would it roughly be the same explanation that you just gave?
Yes. So So the short answer is I wouldn't focus on something being positive or negative too much by itself in isolation. It's always the context of what reference scheme you're looking at. And maybe we come back to it after my next talk, I think, or later today in the breakout rooms.
Then I have we have time for one more question from Janina for Fernando. Could you do an exploratory Loretta analysis with existing ERP data, or does it need a special data set? Does it need a special setup prior to recording?
So if you have, well, the setup would be the recording setup that you have. So if you have sufficient electrodes or on the scalp and clean data, so I would say ideally around 64. And if you have, well, if it's exploratory, you don't necessarily have the brain scan of the participant, for example, but you can use a more general brain scan brain scans that are available to more or less do an exploratory analysis of the ERP distribution on the source in the brain. However, for this remains exploratory. So if you want to have a Loretta analysis, it's better if you have from the beginning a plan to do so, to make sure that you have all the conditions like the 64 electrodes at least, more electrodes better for more precision with the Loretta analysis. And well, also in the cases when you use the scans and also the exact topography or the coordinates of each one of the electrodes, then you can have a more precise estimation of the Loretta.
Okay, thank you very much. We will answer. We had a sudden burst of questions, and we will answer to you, send you written replies. It's time now for our next talk. David is going to be giving us a talk again on EEG hardware and essentially what do you need to record EEG.
Thanks. Yeah, again, we will have time for questions after each talk, but also at the end in the breakout rooms for more complex talks. But yeah, I will talk about the EEG hardware, how it works and how it influences the recording. And first, to give a bit of historical background, no background of historical EEG can be done without Hans Berger, who did the very early or the earliest EEG recording attempts in the 1920s. And back then, there, of course, were not yet any EEG machines. And also the whole methods had to be experimentally proven first. So he also used first participants with trepanation, so with holes in their skull for other medical reasons for better signal quality. Later switched also to a surface level EEG, but still with piercing the skin to really get good signal in. And used galvanometers that were originally not intended also for EEG, but rather to measure currents to find faults in telecommunication lines. And this is his original Edelman string galvanometer from the 1910s. And his technician labeled this photograph as well. You can see, for example, in 1, here is a crank driving the motor. 2 is the straw fiber markers marking the signal on paper. Recording paper switch, graduated lens, and so on. So this is how the first attempts looked like. Again, so the first published publication about this is from 1929. He started a little bit earlier. He was very critical about his first attempts from the 20s, but in 1929 was the first publication. Again, starting with invasive recordings directly from the brain, or with subcutaneous needle electrodes. And one of the first famous rhythms he discovered is the alpha rhythm, because also it's the first that was discovered. Fernando talked a bit about the different frequencies, but this is basically the first alpha rhythm recorded from his own son, Klaus, without holes in his skull, luckily. And on the bottom, you can see here a sine wave, basically noting a time scale. And on the top, you see this alpha rhythm when Klaus maybe closed his eyes or relaxed. So also, as I said, these first recordings were done on paper, so analog recordings on paper. This is a bit more advanced setup already, where you can see this huge machine with multiple channel recordings and a technician doing the recording here. And each of these lines is basically a single channel recorded over time. Also from a very interesting book I can recommend, The Cultural History of Electroencephalography. We won't go into too much detail here because we are not cultural analysts, but I found it very interesting that also in the last 100 years where EEG was developed, it not only had a big impact on science, but also changed basically the culture in various ways. People started thinking about the brain in a different way and about thought and our mind. So it has an impact in a lot of different fields. But again, we will focus on the technical details here. To not overstep our bounds. Again, so 1929, but the first recordings were even earlier. So we also celebrated 100 years of EEG, also in our company, which is why I show this picture here. We have you can find more details about this in our social media accounts, I think, where we visited the Deutsche Museum in Munich, which also has a old EEG machine from the 1950s, like this also very huge machine you can see here. Basically a 2-channel recording, so not many channels. It technically has 2 channels, and so you can see here on the bottom, it's Florian Breitsameter from the Deutsche Museum, Barbara Schmidt from Unic Jena, and our general manager Manfred Jaschke, who basically gone through the inner workings. You can also see here how it looks like these very big tubes that were used as a 2-stage circuit. Of course, nowadays we don't use that big of a tubes and that big of an EEG anymore, but I found it interesting to show you the first inner workings of a properly for EEG designed machine. And speaking about technical abilities, also Hans Berger in his first publication also asked about this. So excuse my German, just to give you the original quote first. Wünschenswert wäre es natürlich auch für die Verfolgung dieser Fragen, wenn zu diesen Untersuchungen noch empfindlichere Instrumente, wie sie die Technik in der Tat herzustellen imstande ist, verwendet werden könnten. So this might also illustrate the very complex German long sentences. I also try to avoid, hope I manage. So I translated it myself to English, and Berger already asked about the following. It would of course also be desirable for the pursuit of these questions if even more sensitive instruments, such as those that technology is actually able to produce, could be used for these investigations. So it's always an interplay between what can be done and what is being done. So the technology innovation also drives the research findings, and vice versa. New research stimulates new technology to be developed. So allow me to briefly show you one example from our portfolio. So the LiFM 64 is modern amplifier from Brain Products, which is small and mobile, as big as a credit card. It's non-invasive, so you neither need holes in your skull and you also don't need to penetrate the skin anymore. So it's much more comfortable to wear. Also, we don't record on paper anymore, but we record a digital signal, which allows also for higher sampling rates and also more sophisticated analysis that cannot be done on paper. And it also has a trigger port for additional communication. So the EEG usually is not used alone, but you want to communicate with other parts of the setup, at least, for example, a stimulation device that shows some auditory or visual stimuli and tells the EEG amplifier when exactly this simulation is happening. To go over the very basic design here, if we have an amplifier, all EEG amplifiers are referential amplifiers. That mean you need at least 2 signals. So you need at least one signal electrode and one reference electrode. And what the amplifier does is it takes the difference between the two, as talked about earlier in the last question. It depends on the exact positioning of these, if you have a positive or a negative signal, but it's basically always the difference between the two that gets recorded and amplified. Then you have an analog to digital converter, or ADC, that takes the continuous analog waveform and represents it in a digital space in a quantized manner, and then gets saved on a hard disk on a PC. It's a very basic design. To put in some more details, you can also split this between a hardware and a software part. On the hardware part, you usually want also to have at least a ground electrode to basically center the recording around a neutral state, have a better signal quality as well. and have some filters on the hardware stage. So you always need a low pass filter or high cutoff filter, which is the same thing. So low pass and high cutoff means it removes the high frequencies and leaves low frequencies in. That's necessary for the ADC to work without any aliasing. I will go into later. And optionally, you might also have a high pass or low cutoff filter, which is the reverse. So you cut out the low frequencies and leave in the high frequencies. And that's sometimes the case, but not always. Then the signal gets transformed to the digital space and to the software. On the software part, you might have an initial step, again, optionally, of decimation, which means you might lower the sampling rate because you might not need the high sampling rate the ADC can produce. And if you have a lower sampling rate than the ADC, there's also the step of an additional low pass or high cutoff filter to prevent aliasing before the decimation. And this is usually all done automatically without any user influence. But on the second part of the software here, this is something you can set yourself in the software. So for example, you can set low pass, high pass, and notch filters. So low pass, again, have the high cutoff, the low cutoff, and the notch filter. Notch filter is a special type of filter that only removes a specific frequency because we have a very prominent frequency no matter where we record, unless we are somewhere in the wilderness, that we have a lot of line noise all around us. The line noise changes depending on the region you're in the world. So in Europe, it's mostly 50 hertz. In the US, for example, it's 60 hertz. And the notch filter removes this specific frequency. And also you have different pipelines. So in the recorder, Lena will tell you more about later, you have at least two pipelines. One is the data that's saved on your hard disk and one that's displayed during your recording. And if you set a filter for one, that's not affecting the other pipeline. So for example, if you want to see a nice signal during recording, you might want to set all these three filters for display only. But for raw data saving, our general recommendation is you usually don't need any filters because once you use these filters and they are saved, you cannot get rid of them anymore. So unless you are really sure about which filters you use, just don't use them. And also, these filters don't prevent aliasing. We in technical support often get questions, should I use this or that filter because of aliasing? That basically does not matter because the aliasing happens in the stages before, and in the stages before automatically employ the correct filters without you doing anything. So the raw data saving filters you set in the software don't save you and also are not necessary. And finally, there's another pipeline. If you have an even more sophisticated setup where you don't only save the data on your hard disk, but you might want to integrate it on real time to some other devices and have a remote data access, that basically bypasses all the software filters here as well. To fill these examples here with some real data for our three amplifiers, the ActiChamp is our amplifier with the highest possible sampling rate, or ADC sampling rate. So you can sample up to 100 kilohertz or 100,000 hertz. It also has two other options, 50 kilohertz or 10 kilohertz. But on the hardware side, you also, in the EEG modules, you have a hard low-pass filter at 8 kilohertz and no high-pass filter. For the brain amp, it's a bit lower, so the brain amp can only go up to 5 kilohertz, and the resulting filters are also lower. There's one option of a 250 hertz low-pass filter, which is mostly needed for special applications if you have combined EEG fMRI, because if you're measuring EEG inside an MRI machine, you have a lot of other signals you don't want in your EEG data, so the strong filter is needed. Additionally, you can also use a 1000 hertz low-pass hardware filter. Also, the brain amp is our only amplifier where you can choose between two different high-pass hardware filter options, DC, which basically means no filter, or a very low frequency high-pass filter of 0.016 hertz. And then the live amp I briefly showed before is a mobile amplifier with even lower sampling rates, but still sufficient for high-quality EEG, either 1000 hertz, 500 hertz, or 250 hertz. And the resulting anti-aliasing filter are around 1/4 of the sampling rate. So 1000 hertz has a 262 hertz low-pass filter, 500 hertz at 131 hertz, and 250 hertz at 65 hertz. And only after this stage, the software downsampling and filters can be employed. So the basic recording principle is that you need a differential amplifier with at least one signal electrode and one reference electrode, and usually also a ground electrode. Then the difference between the signal versus reference electrode gets recorded and amplified, and common mode signals get attenuated in various ways. What are common mode signals? These are signals that are present both in signal and reference electrodes, so basically in all electrodes at the same time, same strength and same phase. And the prime example here would again be line noise. So if you're measuring EEG next to a power transformer, you will have a lot of, in Europe, 50 Hertz noise in all your channels. But it will be at least a little bit, or not a little bit, it will be a lot attenuated, but you might still see something because a power transformer is a lot stronger than anything our brain can produce. But the common mode gets attenuated by the common mode rejection. That's also often a specification for the amplifiers, so the common mode rejection or common mode rejection ratio, which expresses how much these common mode signals are attenuated. One additional option you can use to attenuate these signals is active shielding. This is an option you can use in the ActChamp and ActiCap control box, and it can improve the common mode rejection, but doesn't have to. And tell you a little bit more about it. The channel 1 in module 1 is used as the source for this shielding signal. So if you use this option, you should use channel 1 in module 1 in a neutral spot with, for example, with minimal muscle artifacts. And the way it works is it takes the common mode noise and inverts it and feeds it back to the signal. One example you might know is noise cancelling headphones that work in a kind of similar way. They record basically the audio around you, invert the audio, and basically play you back the audio from your surroundings. And this works, for example, very nicely if you are in an airplane or train where you have a continuous humming of external sounds that can nicely be inverted and played back to you. But if you have a screaming child that fluctuates in its screaming, then it's maybe not as easily. And it's kind of similar with the active shielding here. So if you have common mode noise, so the same signal, the same frequency and same strength in all 4 channels here, flip them, add them to the signal, then you get a perfectly noise-free signal. But if you have non-common mode noise, for example, these 3 channels show the same signal, but this channel does not, you flip it, the average of them, or the channel 1, module 1 source, flip it, add it to the signal, then these 3 noisy channels are now noise-free. But this noise-free channel is then showing the inverted signal you wanted to get rid of. So while you improved 3 channels, you made 1 channel worse, and that might be even worse than not using any of this. So environmental noise can have different characteristics depending on their source. Examples would be AC power lines, computers, display, wireless equipment. And 2 important questions to ask if you want to employ active shielding is this signal present in all channels, and is it similar of strength in all channels? Because the better the noise fulfills this common mode characteristics, the better the active shielding works. But vice versa, the built-in amplifier common mode rejection is often already enough with active shielding leading to overcompensation. Again, this would be an overcompensation. So if you want to use it, I would recommend to pilot your data both in the time and frequency domain and check if it actually makes the signal better. Also, like in all artifact cases, it's even better to eliminate this noise at the source rather than eliminate it after recording or during recording. We will also talk about artifacts in day 3, I believe. And also, as a general rule of thumb, if you're not sure if something makes the signal better or not, do not use active shielding, or rather the law of parsimony. Use the least amount of processing and least amount of fiddling with the data if you're not sure if it really makes it better. So don't use active shielding if you're not sure. Then, if you look at a product page, for example, our ActiChamp, you see a lot of specifications, the sampling rate, the maximum bandwidth, the digitization, input voltage range, resolution, this common mode rejection I just talked about. And one question I also want to answer today is, what are these specifications? What do they mean? So first, to zoom in a little bit about in the analog to digital conversions. So the analog signal is a continuous signal. For example, here I plotted a sine wave that then gets recorded and amplified at regular time intervals, or also quantized. And the higher the sampling rate, the better the resolution. And I simulated two resolutions here. One is 5 times and one is 10 times this sine wave. So either this time is just plotted as 2 arbitrarily, and basically either at 0.2 timestamps or 0.4 timestamps, I sampled these data. And this is the resolving or the resulting waveform. So with the 0.4 time resolution recording, you see the sine wave more or less represented, but blocky. And if you use double the time resolution, it's still blocky, of course, but it's closer to the truth, basically. So the sampling rate depends on the frequency of interest. Our rule of thumb is here to use a sampling rate with at least 5 to 10 times the frequency of the signal of interest. So this would be 5 times the signal of interest, and this would be 10 times the signal of interest, just to show you how it looks. So one example would be if you want to analyze event-related potentials. These are usually happening between 1 and 50 Hertz. So your sampling rate to accurately represent these should be at least 250 to 500 Hertz. Of course, higher is always possible, but this would be the bottom baseline. Also, the time resolution is basically the x-axis, but we also have the y-axis resolution, which is the amplitude resolution. And here it's basically the bits per volt that determine the amplitude resolution. And this also depends on the strength of the signal of interest. So again, having a perfect sine wave here, you can either resolve it at a 0.2 amplitude resolution or 0.4 amplitude resolution. Both will be blocky, but the 0.4 is even blockier than the smoother 0.2 resolution here. So one example here is two examples are the BrainAmp and ActiChamp. This is basically depending on the bit depth of the amplifier. So if the BrainAmp has a 16-bit ADC, which means it has 2 to the power of 16 available steps, which equals around 65,000 steps, while the 24-bit ADC from the ActiChamp has 16 million steps available to it, or 2 to the power of 24. It's also not only the bit depth, but also the measurement range. So basically how big the amplitude can go to be still be recorded, and how many steps you can distribute evenly across this measurement range. For example, the BrainAmp has a 0.1 microvolt per bit resolution option, which means you have 6,000 microvolt possible values, or 6,553.6 to be exact. If you split that evenly between positive and negative values, you can measure from around -3 millivolt to +3 millivolt with these 65,000 steps split between these two edges or boundaries. So in theory, a higher time and amplitude resolution is always better for signal quality. I put it all together here, the 2x2 examples. So again, we have the ground truth here, and the 0.4 time resolution in the middle, and the 0.2 time resolution on the right, and the 0.4 amplitude resolution in this column or row, and the 0.2 amplitude resolution on the bottom one. And of course, on the bottom right here, you have the smoothest representation of the original signal, while here in the middle, you have the roughest. Here would be the intermediate steps. But you also have diminishing returns. So if you double it again, it will be even closer to the truth, but you also need even more storage requirements and also processing demands. So as I talked about in the first presentation, you might have in a general EEG study around 1 billion data points. But if you double the amount of or the sampling rate, you also have not 1 billion, but 2 billion data points. And at a certain point, your storage might run out, or rather, in the hard drives are quite big these times. Your analysis software might not be able to put all the data in the random access memory at the same time, so your analysis might slow down. So the processing demands are usually the biggest hurdle to having a very high resolution. Also, so you might want to stop at a reasonable point, basically. But vice versa, of course, it's even worse if the resolution is too low. And I want to talk about two examples here. One is the measurement range that can lead to clipping. So the amplitude resolution and the bit depth determine the measurement range. This is usually only a problem with a 16-bit BrainAmp because it has only these 65,000 available steps, but can use two different resolution and measurement range settings. So with a 16-bit AD, you have, again, either the option of 0.1 microvolt resolution with a ±3 millivolt measurement range, or you can use a resolution 5 times worse with a measurement range 5 times better. So you can use also a 0.5 microvolt resolution, which equals to a plus minus 16 millivolt measurement range. And if you, for example, then use the low measurement range of 3 millivolt and your signal slowly drifts out of this measurement range, again, this usually does not happen with a pure brain activity, but only happens, for example, if you have slow drifts because of sweating, that can lead to slow deterioration of the signal and also drift it up or downward your measurement range. If it leaves the measurement range, for example, if the sine wave should look like this, if you could record it, but your measurement range ends here and here, you have a flat signal instead of the resolved sine wave. So if you see clipping, you might want to change either your filters or your measurement range, or improve the signal in another way, for example, gel the electrodes again. Another way insufficient resolution can lead to problems is aliasing. So aliasing is when your resolution does not resolve the full frequency of the signal. And one way to illustrate it here is a spatial resolution. For example, if you look at this brick wall, it has a rather high spatial resolution because all these small bricks basically have a pattern that's very intricate. It also depends on how you look at this video at home. So you might also already have aliasing here if your internet connection might not be fast enough to have a high definition version of this talk, but I hope you can see it that this should look nicely resolved. And here on the right side, there are basically not enough pixels available to resolve this high spatial frequency. And the thing you can see here is these artificial patterns, which are basically these artificial stripes that look like they would be in the brick wall, but they are not. They're basically just an interaction of the high spatial frequency and the low sample count in this picture here. This is also called a moire pattern. But we're not looking at brick walls, we're looking at EEG. So an additional example I have here is, for example, in our analyzer, I recorded, in this case, I think it was just line noise, 50 Hertz. And you can see, if I zoom all the way in, you can see this line noise. You can also see it's not a perfect sine wave because the resolution is sufficient to record it, but not perfectly resolve it. So this doesn't look like a beautiful sine wave, but rather like a bit shaky sine wave. But that's not the thing I want to focus on. But rather, if I zoom out here, so if I zoom out, you see this wave gets squished, more and more squished. And again, depending on your resolution, how you look at this video at home, it might also start to flicker already now. So if I decrease first or zoom out in the time window first and then make the window even smaller, you might see flickering here in this signal, and sometimes also lower frequencies appearing and disappearing again, and appearing and disappearing again. It gets more extreme the smaller I make this window, and also the perceived frequency gets lower and lower. So you might see another flickering here. At the very end, you might see a very low signal for a short period of time. So right about now, it should get lower and lower. Yeah, now you see a very low frequency signal that then flips back to a high frequency signal. So this is an example how your display and the zoom factor of your analysis software might also lead to aliasing if you're looking at data. So if you're not sure about some data and you might see a strange signal, it's always a good idea to first make sure you're not having an aliased signal and zoom in a little bit to make sure that's not about the available pixels in your monitor, basically. So aliasing is defined as if different signals become indistinguishable or aliases of one another when sampled at an insufficient sampling rate. Another simulated example here is on the top left. We simulated increasing frequency sampled at a steady sampling rate. And you see these little dots here, which are basically the sampling points. And here's the frequency of this real signal. On the bottom, though, you see the reconstructed frequency based on these sampling points. And first, this follows the real frequency, but at a certain point, it flips. So while the real frequency gets higher and higher in frequency, the reconstructed frequency gets lower and lower because the sample points are not sufficient to resolve the frequency. So if I freeze one frame here, you can see this signal here has a very high frequency, and these sampling points fall in a line that resembles a very low frequencies, because there are not enough sampling points basically to resolve one of these sine waves, but rather they are spread out in a way so you are misled and think that's a low frequency. And the way we solve it in our amplifier design or also software design is with an anti-aliasing filter. And what the anti-aliasing filter does is basically filter out high frequencies that cannot be resolved by the sampling rate or the amplifier. So I have a lot of examples to show it in a different way, but it's an important concept to grasp. So what I simulated here is a chirp or an increasing frequency in blue. You see that the frequency starts with a low sine wave and a higher sine wave, and so on and so on and so on. It gets higher and higher. And then in gray are the sampling points of, for example, the EEG amplifier. And in orange is the sample signal, so the reconstructed signal. And you also see here the Nyquist frequency, at which point this sample signal breaks down. So you might already say, okay, here, for example, this orange line also does not represent the original blue line. That's theoretically true. Let's zoom in a little bit here. That's true that it doesn't represent the full signal, so it doesn't show the biggest amplitude here, but it still has at least 2 sampling points on this sine wave, so it's still the right frequency, even though it might be attenuated. But if you zoom to the right here, for example, this and this is also a kind of a blocky sine wave, but it's definitely not the frequency of the original signal, because here I have 1 sine wave, but in the reality I have 1, 2, 3, 4, 5, 6 real sine waves already happening in this time frame. And what the anti-aliasing filter then does, it suppresses basically the frequency before it's sampled at an insufficient sampling rate. So these artifacts do not happen. I also have an animation now showing on the top the sampling rate or the insufficient sampling rate leading to the aliasing artifact, and on the bottom you see the anti-aliasing filter stopping the artifacts on the right side. So I basically decrease the sampling rate step by step, and you see here on the top these alias signals, and on the bottom basically just a quiet signal without any artifacts. Yeah, this is anti-aliasing filter. So the Nyquist or falling frequency is also defined as half the sampling rate in hertz. So for example, if I have a 10,000 hertz sampling rate, my Nyquist frequency is at 5,000 hertz. And it's the theoretical maximum frequency at which a signal can be recorded without aliasing. However, in practice, only frequencies considerably lower than Nyquist should be analyzed, because you might already see attenuated signals, not aliased signals, so they're not wrong, they're just attenuated and not fully resolved, nearing the Nyquist frequency. And this is another 4 examples showing this here in a simulated manner. So for the 10,000 hertz sampling rate and the Nyquist frequency at 5,000 hertz, which is now more in the region of how EEGs actually work, you can fully resolve a chirp between 0 to 200 hertz, which is, 200 hertz is maybe even a little bit too high. A real EEG signal is usually maybe analyzed until 120 hertz or so. But let's say you have a very, very high gamma, and you want to resolve this signal. You can fully resolve this up to 200 hertz signal with a 10,000 hertz sampling rate. So around here is 100 hertz signal fully resolved, 150 hertz signal, 200 hertz signal, all resolved. If you have a 2000 hertz sampling rate and the Nyquist frequency is 1000 hertz, it's still fully resolved. If we zoom in, you might see some slight attenuation here at the edges, but it's still fully resolved. And if we go to the very borderline of possibilities, if we have a 400 hertz sampling rate and the Nyquist frequency at 200 hertz, this chirp is still technically, all frequencies are resolved, but you see already these big dips before the 200 hertz. So this is the maximum amount of frequency you can resolve, but already before the 200 hertz, you see attenuated signals that are resolved, but not fully. And here on the bottom right would be, if it goes wrong, a 200 hertz sampling rate and a Nyquist frequency at 100 hertz, it only gets resolved until 100 hertz, and then it flips. All these signals on the right are aliased. So the anti-aliasing filter, this is how it would look if you have an anti-aliasing filter. On the top here, you see the aliased or the anti-aliasing filter with simulated data. And on the bottom, I finally plotted a real example, basically just squishing down the signal before any aliasing occurs. And here on the bottom, you see the brain amp with a 5 kilohertz resolution. and a 1000 Hertz hardware filter, and the brain amp with a 5 kilohertz resolution and a 250 Hertz hardware filter. So theoretically, you can resolve signals up until 2500, so half of 5 kilohertz. But in reality, all this part is already filtered out and should not really be analyzed. Only the signals on the left here should be analyzed. And with a 250 Hertz filter, again, for special use cases, because that's usually done in MR, where you have a huge MR artifacts, you see that you basically throw away around 90% of your usable signal just to make sure there's no aliasing and also no MR artifacts in your usable signal, and you can analyze this left part. So I talked about this a lot, and we are always getting questions about people being concerned about this. But I think the important part to remember here is also you don't really have to worry about it. So it's good to know about this, especially also for your later analysis. But on the hardware side, you don't have to worry about it because these filters are automatically employed. So basically, no matter which filters you use and which sampling rate you use, either a hardware or software filter will prevent your signal from being aliased. With the brain amp is our only amplifier where you can actually set the hardware filter in some models where you can choose between 1000 Hertz and 250 Hertz. And theoretically, you can, for example, use a 1000 Hertz hardware filter and then use also a 1000 Hertz sampling frequency and might think, oh, my filter's not enough. But in that case, our software would automatically employ an additional filter to prevent any aliasing, so you don't have to worry about this. Another thing you should worry about, or at least you should be careful about, is impedance, at least one part of the impedance, because impedance actually consists of two parts. One is the electrode impedance, which you can and should influence and make lower, and the other is the amplifier input impedance, which you basically can only influence once you buy the amplifier, depending on which amplifier with which input impedance you buy. And both are important for a good signal. The best signal is basically if you have a high amplifier input impedance and a low electrode impedance. And this relationship determines the quality of your signal. For the electrode impedance, it's also useful to keep in mind that it's not just the electrode that represents this impedance, but basically a layer of things. So first you have your signal source in your subcutaneous layers, which you cannot influence, but you have the skin, sweat, hair, and moisture. That's also a barrier between electrode and signal source. The conductive material, so it could be electrode gel, saline water, or even dry electrodes, and then the electrode type, active or passive, that all get taken together to be the electrode impedance. And the way it usually works is before doing your real EEG measurement, you apply the cap and then apply, for example, the electrode gel and scrub on the skin to make the electrode impedance better, and then measure the impedance, which is done by applying a low voltage alternating current, for example, 15 Hertz or 30 Hertz, which is a bit important to keep in mind because if you would have exceptionally strong noise in this frequency, it can hinder measurement, but that's very rarely the case. And then the opposition to this current is being measured. Just so to clear up the different terms, technically resistance is opposition to alternating and direct current, and impedance is opposition only to alternating current. And because we're using an alternating current, because we don't want to ionize the electrodes, for example, we're talking about impedance but not resistance. But in both cases, we are interested in checking the resistance to this current, and the way it works is the EEG amplifier applies this current to some sort of input voltage electrode. This depends on the setting you're using and the amplifier, but let's, for example, imagine you do that via the ground. Then the current flows through the conductive material, the electrode gel at the ground position, the skin, sweat, hair, and moisture at the ground position, through the skin, sweat, hair, and moisture until you reach the signal electrode, where the signal electrode gel is measured, and then finally the electrode itself is measured and it flows back. So it's important to keep in mind that this measurement is always a loop involving at least two electrodes at the same time and always having the ground and reference in mind, because the ground and reference need to be basically at least attached and also as low as possible for this measurement to be accurate. Because if the ground would not be put on the head, or maybe it has fallen off, you don't get any useful signals out anymore because this loop is not closed. Also, it's important to note that the impedance measurement is only an approximation within the impedance measurement range. So it's not to the last digit perfectly accurate, but it doesn't need to be. It's basically only an approximation for us to know, is this electrode good and can I measure good EEG signals? So the impedance by itself doesn't help you. It only helps you as an indirect measure of the signal quality you might get from this electrode. And the reason of this approximation is because we are applying a low voltage current. Also for safety, basically, we don't want people to be shocked, and also we don't want them to be feeling anything, so they don't feel a tingling on their skin if the measurement is too strong or the voltage is too strong. It's enough to apply a very low voltage, so no one is hurt, of course, and also you don't feel this measurement going on. There's also two distinct electrode types I want to talk about here, is passive and active electrodes. Because for passive electrodes, one important step is also the electrode cable, or one unfortunate step is also electrode cable, because the measurement of both the impedance and also the EEG data does not distinguish between electrode cable and electrode. So the electrode cable can act kind of like an antenna, recording all sorts of environmental noise if you're in a noisy room, which is added to your EEG signal you are interested in. But if you use an active electrode, there's already the impedance conversion happening at the location of the active electrode. So the electrode cable is actually not a big issue anymore, and no cable noise or minimal cable noise is added to the signal because of this active electrode technology. And this also serves two purposes. For one, it prevents the cable noise, but it also makes it easier to prepare and use the electrode, because you can see the impedance already in the LED of the active electrode, so you don't need to cross-check with a monitor that might be hard to reach, and you have to mentally calculate this electrode on the screen goes here. So the preparation is faster. Laura will talk more about this, I think, later as well, about which electrode technology to use. But in general, with active electrodes, you have high electrode impedance that and higher environmental noise possible without a loss of signal quality. So you can get away in a worse environment and with a worse preparation and still have a good signal quality. To go over some impedance threshold examples here, for the active gel-based electrodes, ActiCap, we recommend being lower than 25 to 60 kilo ohm. For the passive gel-based electrodes, it's a bit of a point of discussion, because a lot of people also still write us, for example, in a paper by Luck from 2005, he recommends impedance threshold of very low, below 5 kilo ohms, while other studies also found that with modern amplifiers with higher input impedance, which all our amplifiers have, you can get away with higher impedances also below 40 kilo ohm with a similar good signal quality. With a passive water-based electrodes, it's even higher, 30 to 150 kilo ohm, and the highest one would be active dry electrodes, so without using any gel, which can get away with up to 2,500 kilo ohm. Yes, and important to note, as reported also in this committee report by Kyle and colleagues, the amplifier, the electrode impedance is less important than the relationship between electrode impedance and amplifier input impedance. So I briefly touch upon this as well. To give you some example values, the ActChamp has 10,000 mega ohm, so not kilo ohm, but mega ohm input impedance. The BrainAmp standard 10 mega ohm, and the DC can even switch to 10 giga ohm. LifeHand similarly 200 mega ohm. And importantly, as I said earlier, the active electrodes technically have an input conversion already happening in the electrode. So you can also think about this as an input impedance. And the ActiCap has an input impedance also around 200 or higher than 200 mega ohm. Again, Laura will talk more about what to choose for your setup. I just want to briefly mention the basics here. Because another part influencing the impedance is the skin we all hopefully have, and we don't need to pierce all the way through the skull anymore, like in the olden times. Yeah, and skin can have different properties depending on what's on it and what has been done with it, basically. So exposed skin has around 200 kilo ohm of impedance, and protected skin, for example, when there's still hair on it, has around 120 kilo ohm. That's also one reason why bald people are actually not the best EEG participants. Oftentimes they say, oh, I'm perfect, I don't have any hair. But any hair, don't have any hair is actually not the best. Also, having a lot of hair is a bit hard because you have to get through all the hair. So the best part is also if you're slightly balding like me, that's perfect. I only did it for the EEG, but that basically reduces your impedance, and because you don't have too much hair, you also have a easier way to reaching the scalp. But of course, it's not only the hair you can hardly influence. You can have to get used to what the participants have. But you can and should definitely encourage them to come with washed hair. And or if that's not enough and still grease on the hair or the scalp, you can apply some alcohol under the electrode to scrub away some of this layer. You can also get even lower impedance if you carefully abrade the skin. So it doesn't mean you have to be, or you should not abrade it until it bleeds. So just gently removing the uppermost layer, for example, with abrasive gel that has some small nodules in there, which basically scrub away a bit of this, or a blunted needle, not a sharp needle. And it's also important to note it's not always needed. So only if you really need low impedance, for example, for special use cases in the MR, it's very important, or TMS. But if you use active electrodes, that's usually not needed. There also are different gel options depending on your needs and setup. So one is clear, which is only conductive, and the other is abrasive, which is both conductive and abrasive. There's liquid and viscous, and there's salty and free of salt. So basically, the recommendation for active electrodes is it's usually enough to just use clear gel and no abrasion. If necessary, you can do, but usually you don't need any abrasion. For passive electrodes, though, we recommend using abrasive gel, which has some gentle abrasion properties already, because passive electrodes, again, are a little bit less, or you need a lower impedance to get a good signal, usually. For liquid versus viscous, there's basically also a cutoff depending if you have a low or high channel count. If you have a low channel count, you might get away, or you might want a medium or lower viscosity because it flows easier through the electrode and also to the skin. But the trade-off is you also have the higher probability of having ridges, so not only flows easily to the skin under the electrode, but flows from one electrode to the next electrode if you have two electrodes close by. So if you have a higher channel count, we recommend using a higher viscosity gel to prevent the gel from forming bridges. Salt versus salt-free. It's also a thing that the skin, so depending exactly on how your skin is and if you did sports or something, but usually the skin has around a 0.9% salt content, and the salty electrode gel is considerably higher, between 3 to 10%. And this leads to a self-improving impedance over time because the salt wants to equalize this gradient, and basically they exchange the salt between each other, between the skin and the gel, which makes the impedance improve. That's also part of the reasons why you should not focus on one electrode too much during preparation. If you say, oh, my electrode FC1 is bad, and then spend 10 minutes just on this one electrode, just prepare it a little bit, scrub a little bit, put a little bit of gel in it, and then go to the next electrode, because by the way, by the time you might have prepared all electrodes, the salty and non-salty skin might have exchanged some salt, and the impedance might have improved without you doing anything. That's especially the case for slow potentials. So if you're interested in DC recordings or below 3 Hertz data, I think Fernando talked about like these late potentials for language, doing wrong language with the correct language. You might need these preparations. But on the other hand, salt can be unpleasant, especially if you use it with children and or abraded skin. They might be a bit more sensitive. So you can also use salt-free gel. Then some words about the electrode cap layout. So there are different layouts, how you basically distribute your sensors around in the scalp or on the top of the scalp. But the most common one, also our standard electrode system, is a 1020 system. And it's called 1020 system because it consists of connecting lines that are divided in 10 or 20% increments. The way it works is you have 4 landmarks. So the nasion, which is the deepening between the nose and the forehead. The inion, which is the lowest palpable rim at the back of the head. So you usually can feel it if you just put your finger on the back of your head and then slide down until you feel this rim. And the left and right ear channel opening, or usually pre-auricular points, so before the ears, but basically the ears are another landmark. Then you connect all these four landmarks and divide it in 10 and 20% markings. The first 10% increment is basically the headline. So nowadays, not that many people wear hats anymore. Maybe that was more back then, but I don't wear hats. But anyway, if you would imagine, that's not correct. If you would imagine you wear a hat that's around the headline, is basically where you put your first line of sensors, and then every 20% the other one. And the resulting points are approximately equidistant, also across participants. Again, there are differences also in the head shape and so on, but that's how it works. But it's important to note that this 1020 system originally only has 21 electrodes. So this is one example layout with the 21 channel layout, where you have these equidistant electrodes together with on the ears and the ground and ref. But most of our setups have an increment of 32 channels, so either 32, 64, and so on. And the way it works is that we take the 1020 system and fill it up with some electrodes from the 1010 system. So the 1010 system is basically doubling the amount of electrodes by not only using the 20% landmarks, but also the one in between. So this would be one 32 channel layout we provide. And also we cannot fill all 1010 systems because it's around 70 something electrodes, which again does not fit nicely with 32, 64, 96 channels. So we basically fit it out in the regions that are usually most interesting for EEG. So around the center of the head and on the back of the head. And if you go one step further, 64 channels might look something like this. So we also fill in the gaps here. You usually also need then 2 electrode sets. So that's why they're colored differently to fill it out. And just to show this as well, if you go all the way to the maximum amount of channels, the 256 channel is possible, for example, with the brain cap. You have a lot of channels, so filled up, filling the gaps once, twice, thrice, and so on, and a very high channel density. But it's important to note that's not necessarily better for all use cases, because you need, for one, you need more money to buy it, but you also need more preparation time. You have a higher risk of bridges and so on. So it's not always better to have the highest maximum amount of channels. So to sum up, EEG, electrode, nets, caps, gel, many different caps exists, active and passive, gel, wet, dry, high channel count, low channel count, for special environments, for the MR, MEG, TMS, sleep. We also provide fixed or variable reference electrodes, where for the ActiChamp you can use a different electrode if you want. For the others you have a fixed electrode. Reference electrode. Also, the electrodes itself are sometimes fixed into the cap, and sometimes you can also remove them. Different sizes for adults and children. Different head shapes, oval cuts and round cuts. That also differs between, for example, Caucasian and Asian heads on average. Again, each head is different. And different fabrics, so either optimized for high precision or high comfort. And some caps might also have auxiliary sensors already built in, for example, for eye activity, muscle activity, heart activity. And it's important to note there's no perfect setup. It's always a trade-off between signal quality, ease of use, and preparation time. And putting it all together, I think she was not yet here, so I still have time for my last slide. Putting it all together, you usually want to record various things from the person. So you might want to record the response, the brain signal, and peripheral physiology. For example, for the response, you might want to present stimuli A and B with the response, maybe even also left and right hand, and send triggers to the EEG amplifier to record when these responses and stimulations happened. Then on the sensor side, you might also want to record, of course, the EEG signal with a reference and ground to the EEG amplifier. And additionally, you might want to record peripheral physiology, for example, heart activity and breathing. And all this then gets recorded in one amplifier and sent to the recording software during recording, and afterwards in the analysis software, where we will talk about analysis later on the next days, to analyze all these data in a meaningful way. And with that, I'm finished. So I have time for questions.
Thank you, David. It was covered a lot of ground, and I'm sure a lot of it would be very valuable for attendees. Just to get things rolling, maybe ask you a first question. Question from Subhashini. Just as we can see that what is the typical minimum sampling rate that would be good for EEG measurements?
So I would say the minimal typical sampling rate would be around 250 hertz or 200 hertz, because I would say the lower frequencies you're interested in is usually event-related potentials. If you want to look at these, you want to look at least 40 to 50 hertz signals. So 200 to 250 hertz, like 5 times that would be the minimal sampling rate. You theoretically can go lower sometimes, but you should really think about what you want to do then.
Okay, yeah, that makes sense. Another question is, could you clarify a bit more about this, the difference between passive and active electrodes?
Mm-hmm. Yeah, so one important caveat maybe also to be to weigh both sides is, because I'm of course we would recommend for most use cases active electrodes are our best go-to solution. Again, Laura will go into more detail later, because they are easier to employ. You can see the impedance already on the electrode. You are more noise immune to environmental noise and can get away with higher impedance, so you might not need as much preparation time. But on the other hand, if you have in a perfect world, basically, if you have a shielded room, you have a lot of time to prepare the sensors perfectly, and don't have any environmental noise, passive electrodes can technically still be better because they don't have this additional step. Just like in real scenarios, for most use cases, the active electrodes have a better immunity or better trade-offs, basically, for all the environmental sources. And finally, for active electrodes, there's also they have to be treated with a bit more care. Again, I'm working in technical support, not in sales, so we talk a lot about problems. But one common problem we have with active electrodes is, because they're active and they have some circuit boards in the splitter box, this splitter box sometimes gets wet, and it must not get wet, because water on the splitter box basically damages the electrode set to the point of non-repairability. And that's another trade-off of active electrodes. They are better, more immune to noise, but also and easier to handle, but also you have to be a bit more careful to not damage them. And in a perfect world, passive electrodes might still be better if you have the time and environment to use them.
Okay. Here's another somewhat related question from Tatiana. If someone's using sponge-based electrodes, how would you decrease impedance? Because I guess there is no gel involved, right?
Mm-hmm. Yeah. So first, for water, it also has to be said, it's not just water, it's saline water, so you have to drench it in salt first. So the salt is actually the one that carries the current. There are various ways to increase or decrease the impedance. First is, of course, when you prepare the electrodes, you want to make sure that the electrodes are properly drenched in the saline water. And for another, you can not with a gel dispenser, but rather like a little water droplet, put some water under each electrode. If that's not sufficient, again, you can use the cleaning we talked about before. So tell the customers, or not customers, participants, to arrive with washed hair, maybe even degrease slightly with alcohol if needed. And then another trick can be to brush the skin. So not the it's not about brushing the hair, because the hair will get messed up by the electrode anyway, but basically use an anti-static comb. It's also important, not a static comb where your hair stand up afterwards, but use an anti-static comb to basically roughen up the skin a little bit, because with this roughened up skin from the mechanical irritation, it's a bit easier for the gel or water to flow into the cracks in your skin to make a good contact with the electrode. And finally, especially for the RNet, one trade-off, or I would also say one benefit of the sponge-based electrodes is they are faster to prepare usually, because the water just flows more naturally in the on the head, and it's also a bit easier to wear and wash off later. But it also dries out a little bit faster. Gel stays good basically for longer, and water, depending on where you are, if you're in a very hot and dry area, it might dry out already after like 40 minutes or so. So if you have a very long experiment, like longer than, let's say, 1 hour or so, you might want to include a break in your experiment to check impedance if something dried out, and if something dried out, then come back in the experimental room and wet the sensors a little bit.
Hello, everybody. Welcome back. We continue on with our program, and our first speaker after the break is Laura, who will be telling us about how to choose the right amplifier and electrode technology for your research. Take it away, Laura. It's all yours.
Thank you, Shiva. I'm trying to share my screen. Let's see if this works for you. Okay, you can see okay? Then thank you everybody for joining and for hanging in there with us. And I hope you're a bit fresh after the break. You had some time to get a coffee. We will continue right away. So we have heard a lot about recording EEG already, about the background, the hardware. And in this very session, I will focus on choosing the electrodes and the amplifier for your own research. And as David already presented, there's a lot of variety, different technologies, and this can be challenging because we have quite a variety of options to choose from. And this session is designed to help you find out which of those options is the best for your own research question. So to start with, let's take first a closer look at the electrodes. So David already mentioned a bit about the technology. So what all scalp EEG electrodes have in common is that they have to make contact with the scalp. So we place them on top of the scalp and we try to detect electrical potentials that originate from brain activity. And the more we improve the contact between the scalp and the electrode, the better this electrical signal can pass through from the head to the electrode. And what hinders or impedes this current flow is called impedance. And I believe David already explained it. You could call it resistance, but impedance is the correct term for the resistance to alternating current, not direct current flow. So, as David mentioned, there are different electrode technologies. One major difference between them is how they establish the contact with the scalp. And this can either be abrasive gel or viscous gel, as you mentioned, or it can be sponges that are soaked in a salt water solution, or it can be directly against the skin without any conductive material. Those are called dry electrodes. So we differentiate between gel-based, sponge-based, and dry electrode systems. Now, resulting from this difference in how they work, how they make contact with the scalp, we have to expect different impedance levels. So basically the target with impedance, which impedance we want to work with, is different for the gel-based systems than for the dry electrode systems, for example. I've put some numbers here, what is roughly considered as the goal impedance value for these different technologies. And here, remember, lower impedance means better contact with the scalp and potentially more signal arriving at the electrode, whereas high impedance means that we detect potentially more noise and less signal. So with gel, you can get down to basically 0 kilo ohm impedance if you really want to. With water, it's a bit more difficult. So salt water has some conductive properties, but you still have to accept that you work at 60 to 100 kilo ohm impedance values. And for dry electrodes, we have to accept even more impedance, even stronger impedance. However, if you have set up different electrode systems in the past, you will have noticed that it takes a lot longer to set up gel-based electrodes. You have to prepare every single electrode head with the correct kind of gel. You might have to abrade the skin, and this can be a real disadvantage because it requires time, and you might not have this time. So for dry electrode systems, this is the advantage. And then also briefly mentioned before, if you want to do long recordings, you don't really have a choice. You might have to use gel, because salt water will dry out after maybe roughly an hour, and the dry electrode systems work for a longer time, but they have to apply pressure to the skin, so they become uncomfortable after a while. So here you would have to also face this trade-off of how long you can record with which technology. And then talking about electrode comfort, this is probably best somewhere in the middle, where you might either you don't need gel, or at least not abrasive gel, and you also don't work with pressure on the head. So for the participant, it's probably most comfortable to work with the sponge-based systems, or at least with active gel-based electrodes. Okay, so it always looks like we have this strong trade-off between the good signal-to-noise ratio and long measurement durations versus convenient quick setup and clean hair, comfort for the participant. However, this trade-off is a bit less extreme than it looks like here, because there are things we can do, especially concerning signal-to-noise ratio, comfort, and preparation time. So let's take a look at these 3 points. A few things have already been mentioned. So first of all, our amplifiers all work with high input impedance. So as already mentioned, electrode impedance isn't everything, but what counts is the ratio to the amplifier's input impedance. And all of our amplifiers work really with sufficiently high input impedance to improve your signal a lot. Second, we have also mentioned the active electrode technology. So you have to imagine that the electrode is like an antenna that picks up all sorts of electromagnetic activity. And this is not just referring to the electrode head, but also to the cable. And now, if we use active electrode technology, every single one of these electrode heads has a tiny converter that performs an impedance conversion. So whatever happens to the cable afterwards does not really affect the signal that much. So here maybe again illustrated why active electrode technology can be so good for reducing noise and improving the signal-to-noise ratio. Then another thing that is applied by our dry CGX electrode systems, for example, is to shield the electrodes and cables, specifically with dry electrode technology. So these dry electrodes have a small pod that encloses every single electrode head, and it's like a tiny Faraday cage for each electrode, and also the cables are surrounded by this plastic cover, and that makes sure that noise does not affect the signal so easily. And last but not least, another trick is to route the cables really tightly along the cap. So even if you work with passive electrodes, the noise is not going to affect your cables that much. You remove noise introduced from cable motion, which also improves your signal-to-noise ratio. Okay, this is what we can do about the noise problem. What can we do about preparation times? So yes, you might have to work with gel-based electrodes sometimes, but we have integrated these small LEDs into our active electrode heads, and they indicate directly at the electrode how good the impedance is. So you can basically set your threshold yourself, which impedance level you consider as good, and then you can see directly during preparation at the cap if the impedance is still too high, getting better, or turning green if you are under the indicated threshold you want to be at. And this is also implemented for our dry electrode systems. So this makes it really, really fast to set up the caps and to see right away where the impedance is maybe not optimal yet. Then another point that you need to consider is that active electrode technology means you can accept higher impedance levels. So instead of working all the way down to 10 kilo ohm, 5 kilo ohm or lower, you can just accept higher impedance values like 25 kilo ohm, for example, for the active cap electrodes. And that saves you time because you don't need to spend so much time on electrode preparation and the signal is already fine. And then talking about electrode comfort. Of course, again, accepting higher impedance values means you don't have to abrade, you don't have to rub, you can just use sponges. And another trick is to shape the dry electrodes in a sort of a spider shape. So it's not just one pin pressing against your head, but the pressure distributes and the electrodes can make contact with the head, and then you can wear them for quite long before the pressure becomes unpleasant. So these are some hacks, and we try to apply a lot of them for our solution, so basically as much as possible. And this makes this trade-off a bit less critical, as I mentioned. And this means that basically different electrode technologies could be used for a variety of situations, research questions, but we still want to help you to find the best solution. So what I want to do is to go through several questions, and depending on your answers, one or another system might work better for you. I will do this for the electrodes first, and then I will do the same for amplifiers. And in the end, I also want to see if there are maybe some special research applications that you need to look out for that have a clear indication which electrodes, which amplifiers you should use. So the very first question I want to ask you is, what are the requirements on your data? What are the requirements on your data analysis? What is your goal? So having useful data for you is most likely the primary decision factor, because if the data is not usable for you, you basically wasted your time and you cannot publish. So maybe your analysis goal could be straightforward, and reaching it might not depend on having the highest signal-to-noise ratio. This might be the case for event-related potentials, because here you average usually across hundreds of bins, and then the noise will just cancel out naturally and a signal will come through. This might be a case where your requirements on your data are not as crazy strict, or you might be interested mostly in medium range frequencies, let's say between 1 and 45 Hertz. These frequency ranges are not as affected by noise as are extremely low or extreme high frequency ranges. So if your analysis goal looks something like this, then you can choose your electrodes freely, but in this case, it might work for you to accept higher impedance levels, and you might be happier investing into gel-free electrodes with, let's say, sponge-based electrodes or dry electrodes, because this means the setup will be quicker, you can gather data from more participants, you have maybe more time for collecting more trials, and you can just boost your statistics by collecting more data, and you don't have to dig so much into getting the lowest possible impedance and the craziest, best signal-to-noise ratio. The systems we have for this are dry or sponge-based. The RNet on the left side, this is the one with the sponges, then the ActiCap Express Twist with small pin electrodes, or the CGX Quick system on the right side. However, maybe your analysis goals are more difficult to reach, so maybe more complex analysis. I just put 2 examples here, don't take them as the main reasons to use a different system. But for example, if you're interested in very low frequencies or very high frequencies, or if you need a very strong signal-to-noise ratio because you are really relying on few trials, or because you are looking for an extremely small effect, then maybe you need to invest some time into gelling your participant's head, into preparing every single electrode, and to getting really low impedance levels and better signal-to-noise quality to start with. And there our solutions are the ActiCap Slim or ActiCap Snap on the left side. This is the one with the active electrodes, or if you prefer the more, let's say the more traditional passive electrodes that are equally good, then you can use the BrainCap. Okay, there might be other analysis goals you might have. For example, do you want to perform source analysis? Or do you want to extract independent components from your EEG channels and work with them? Or do you maybe want to use an average referencing scheme and look at, instead of using the reference behind the ears, for example, look at the whole head as a reference? Then these are all good reasons that you need a higher coverage, that you need a lot of electrodes and not just a few. And in this case, we recommend you to record a minimum of 64 channels. And in this case, the first thing I would recommend would be a sponge-based system like our RNet, because you can just soak the whole net in water, and then you dip it dry a little bit, you put it on the head, you wriggle around a little bit, And this should be good to go actually. So this will save you insanely much time as compared to preparing every single electrode on the head, especially for high coverage. And as you can see here, the RNet can also cover the whole head with up to 160 channels. So there you have covered the whole sphere of the head, which is also helping for source analysis to make it more reliable and more correct, let's say. You can, if you really want to have high coverage with gel-based electrodes, if you need a fantastic signal-to-noise ratio, you can work, for example, with the ActiCap electrodes. Here you can also cover the whole head. In this case, is actually still relatively fine to set up quickly, and you can work with the CapTrax localizing system. So this means you can map these electrodes due to the LEDs. You can map them to, for example, an MR scan of the brain, and you can precisely adjust your analysis to the participant's brain you're working with. So here again, fantastic for source analysis of your signal. And as I said, still relatively fast to set up because you have color-coded holders. So you can always insert 32 channel bundles into one specific color and then advance with the next color. And you have the LEDs, so when you're reducing impedance levels, then you're still relatively fast because you can just use the little lamps. Okay, so let's say you have asked yourself the decisive questions about your analysis goal, and you have found out if you have complex analysis that needs good signal-to-noise ratio, or if you have indication for high coverage or not. The second question I would ask is, who are your participants? So, for example, it could be that you're working with a sensitive population. This could be infants, for example. This could be patients who are already not feeling well and who cannot spend too much time on electrode preparation, or people who are scared of syringes, who don't want to have any skin abrasion. So in this case, electrodes that can avoid gel or can avoid at least abrasive gel are better for you because you'll have more compliance from your participants. So for child research, the RNet is really the perfect solution. We have infant sizes that go really also down to newborn babies, and they have very soft straps of silicones that can be used to fit to different head shapes, which is also important for babies. And they don't work with syringes at all. As I said, you just put on the net and it should be fine, but you can work on still work on decreasing impedance values with these tiny pipettes and add a drop of water if needed. Still, if you need a really good signal-to-noise ratio and you have a sensitive population, the ActiCap is also an option. So this can also be used in children because, again, the setup time is relatively fast. If your participants really have no time at all for a full-blown setup, or they don't want to get their hair dirty because they are still at work or they are in school, or you're talking to important people who don't want to get their hair dirty, then this is an indication that you should work without gel just to get enough participants to actually come and participate in your study. And so both the dry systems like the CGX system or the RNet don't make the hair dirty, just mildly humid in the case of the RNet. And this should probably improve the participants' will to participate in your study. Okay. And then one last question concerning your participants could be if they have various head sizes. This is a factor because in that case, you would want to work with systems that you can use for different head sizes, not just for one. And you don't have to buy a single electrode set for every single head size. This can get a bit expensive. So here, the ActiCap is quite optimal because you have independent electrode bundles and fabric caps, and you just snap the electrodes into the cap holders. So basically, you can combine any cap size with any electrode bundles. You don't have to buy more bundles than you absolutely need. And another option for a quick setup without getting the hair full of gel, you could use the CGX Quick system. So this fits sizes from 52 to 62 because these headsets are kind of bendable and you just pull them on the head like arms that kind of press against your scalp, and they really adapt to the size and the shape of the head. Okay, so we have looked at our participants. We have found out if they are particularly sensitive or if they have little time or varying head sizes, and then can answer which electrode we want. Now let's take a look at our amplifiers. There's again quite a variety of solutions. I have depicted 4 of these solutions here. I just want to rephrase shortly what amplifiers do, not to explain it to you, this has already been done, but to tell you how these amplifiers differ and why this might matter for your study. So as the name says, the amplifier amplifies and digitizes the EEG signal, usually with the help of the ground and reference and the signal electrodes. You have different sorts of amplifiers. The signal electrodes are always first referenced to the ground electrode, and then to improve the signal quality, you can look at the difference between signal and reference electrode, a dedicated reference electrode, so that it will look cleaner, and you can work with that. And differential amplifiers really amplify the difference in signal between signal and reference electrode, not between signal and ground electrode. A bit oversimplified. So this is what most of our amplifiers look at. Then we already mentioned several hardware filters that are there, and then importantly the analog to digital converter. So how do we convert this continuous signal into discrete digital values that our computer can handle? And here also David mentioned the sampling rate or the sampling interval is decisive. So how many values can we depict, can we record per second? And the resolution, how many values can we look at on the Y scale, like how finely grained is our recording resolution, and how big is the range? So when will the signal cut off because it becomes too large or too small? And this all determines basically the properties of our amplifiers, together with the option to add trigger signals, so to mark events in your EEG data, and also to align sensors, so peripheral data like heart rate or EMG to your EEG signal. So these are potentially things in which amplifiers differ. We have a document online in the scope of also a press, like an article that lists all these technical features of all our amplifiers. You can look up if you have very specific questions, but I want to give you an impression how they mostly differ, which might be relevant for your first gut feeling what you need. So the first thing, of course, is the sampling rate. This means, again, how many data points you can record per second. And there clearly the ActiChamp Plus is the strongest one. So here you can record 100,000 values per second for 32 channels. You have to consider that the maximum sampling rate means also you might oversample, you might record too much data, you might have to be careful if you record a lot of channels, then you might have to reduce a bit on the sampling rate just to not overwhelm your recording or analysis program with data values. But here you can already see that the bigger amplifiers have potentially stronger, more powerful options to sample, and the smaller ones that are more mobile have a lower limit how much they can sample. Then there's another factor, how many channels you can record. Basically, almost all of our systems, or like all of the stronger brain products, amplifiers, can record 64 channels or more. The smaller mobile systems, sometimes less. The ActiChamp can record up to 160 channels, and then the BrainAmp on the very right-hand side. This can be combined with up to 8 modules to record 256 channels. So this is a really high limit. You might barely ever need this amount of coverage. And then on the other hand, another thing that is important is the battery life. Here again, caution, because you can extend the battery, for example, of the live amp on the very left side. You can extend this with a power bank. You can swap batteries if needed. But in principle, again, the big amplifiers have stronger batteries that last for really, really long, that can be used for overnight sleep recordings without ever exchanging the battery. And then coming with this property, also the bulkiness will increase. So the smaller mobile amplifiers with the lower specifications are much lighter. So the live amp, for example, would fit into the palm of my hand, whereas the brain amp, as I already said, might consist of more than 3, 4, 5, 6 of these boxes that are depicted. So it becomes really bulky and really hard to move. Okay, so there's not such a clear trade-off for the amplifiers. They just have very different properties. So the main question I would ask you is, do you need a lab setup or do you need a mobile setup? Because this comes with fundamentally different requirements on the amplifier and on what you can do with it. You will probably all know this standard scenario. In the lab, the participant is sitting in front of a computer. You don't mind the cables. You can have basically the setup as large as you want. They are doing their task on the computer. But now with the technological advances and with more challenging research questions, we get the study to really go outside to the field and to look at naturalistic conditions. So here we're looking at completely different scenarios. So no cables, no mains power that you can use, and more motion. So basically, to highlight the main differences, in the lab you will have larger, more powerful setups that can be extended to have more triggers, more cables, more inputs, while the mobile setup has to remain small, it has to remain light, and you have to be able to carry it around. Yeah, the lab setup is potentially bulkier, while the mobile setup cannot afford to be so. Then on the lab, you can use cables to transmit data. This is a very safe way, a very fast way, whereas mobile systems are often dependent on wireless data transmission via Bluetooth or Wi-Fi. And then the lab setup functions perfectly in noise-free environments, but the mobile amplifiers are really meant to withstand motion and noise. And then what you can measure, of course, is maybe less naturalistic conditions versus very naturalistic conditions in the mobile setup. Okay, so for the lab setup, if you go for that one, because this is what you need, then we have mainly two different big lab amplifiers. The more compact and more powerful solution is the ActiChamp Plus. So this is our all-rounder, our latest development of lab amplifiers at the moment. So here you can still have a portable system because you can add up to 5 of these 5 electrode bundles of 32 channels, so you can have up to 160 channels, and it still stays portable. It's still kind of compact and can easily be carried from room to room. And you have also 8 integrated channels for additional sensors. So if you want to record EMG, EOG, heart rate, pulse, whatever you want to add to this, this is possible. It's no problem. In the back of the amplifier, you have an 8-bit trigger port that is already integrated. And yeah, basically the main advantage of this system is very powerful while remaining portable and remaining quite compact. The other possibility could be that you don't mind a large system, you don't mind this modularity, but you need other specific features. So we have the brain amp here. And this comes with independent 32-channel modules. So each of these, maybe you can use actually my pointer, that will help a bit. These boxes can handle 32 channels in the front in the slots, and you can stack up to 2 of them on one battery, and you can record up to 8 of these 32-channel modules together to come up to 256 EEG channels. And the data of all of these modules is integrated in this USB adapter box. And here comes also the advantage of this apparent bulkiness or this apparent huge modularity. So this USB adapter synchronizes the clocks of any attached amplifier modules, while you can still treat them as separate instances and the devices are galvanically isolated. So this makes the brain amp actually our best or cleanest solution for hyperscanning with EEG. That means you can connect 2 different people to 2 different amplifiers, but synchronize them with one USB adapter and have all the EEG data aligned perfectly on the same computer clock. If you want to send trigger signals, this will also be integrated directly in the USB adapter. And if you want to add sensors to your EEG recording, that would be in separate modules. So BrainAmp EXG can record up to 16 different sensors that can also be added. And also here you see more sensor inputs up to 16, triggers also up to 16-bit if you need. So this is a bit bigger if you need it. Okay, now having taken a look at the lab setup, what if you want a mobile setup? Exactly, here basically you decide between the features that are most relevant for your lab setup, but the mobile setup, the main question I would ask is how strong is the motion you expect? Is walking involved? Is even running involved? Is it during sports, or is it just slow? So if it's a mild motion setup, if you just maybe take your mobile amplifier to the field, you put it on someone doing their daily job, sitting on a desk, then there's absolutely no problem to work with the CGX system, so with the dry electrode headset, because you can set it up quite quickly, you can take it off quickly, you will not have any dirty hair. Here the amplifier is integrated and the headset is on one side, like on one ear, while on the other ear you have the batteries integrated. And also here you have the option to send wireless trigger signals, so you can also mark events in your EEG data. And you also have the option to record sensor data, although they will not be aligned to the EEG data, there will be a separate recording instance. So this is for mobile recording with a mild motion, let's say, or without motion. If it should be handling stronger motion, then we would recommend the live amp. So this is this tiny amplifier of 60 grams that fits into your hand. Optimally, it would connect to our ActiCap, so the active electrodes with gel, and this solution is really the one that withstands noise the best. You can extend it to 64 channels simply by clipping 2 live amps together, and it will treat the whole recording like one 64-channel recording. So you can really use this sort of setup for more challenging situations. We've had people using it for bungee jumping or for parabola flights. So this is really our most noise resistant system in combination with the ActiCap. You can extend the live amp. So the live amp itself only has a 1-bit trigger input, not way more, but you can extend it with this sensor and trigger extension. So connecting up to 7 different sensors, and also receiving wireless triggers here. So a wireless trigger receiver is inbuilt into this device. And the whole setup would look like here, still very compact, can be just attached to the participant, and then they can do their sport or whatever you have planned for them. Okay, so for the mobile setup, mainly my question would be concerning how much time you need to set it up, how long would you want to record, of course, and then if it should be noise resistant or not. Okay, now the last question I have left to ask is, are you recording under special conditions? Because electrodes and amplifiers, nice, mix and match, but there might be conditions that really bind you to one solution or the other. This means, for example, you might be measuring EEG in a challenging environment, or you might want to combine EEG with another neuropsychological technique, so this might also interfere with the EEG recordings. We have already touched on some of those solutions. Here's just listed a few that you might be interested in. We have looked at infant research, so here again, you choose whatever you like, but for example, the RNet might be beneficial to be as fast as possible, not harm the kids, not scratch the skin at all. And for sport, I already mentioned the live amp and the ActiCap electrodes that will just give you the best solution or the best data. And then for hyperscanning, this in principle works with all of our amplifiers in one way or another. But for example, the BrainAmp would be the cleanest way, where everything is really aligned on the same computer clock. I want to touch on a few of those in a bit more detail, namely those that have kind of specific requirements. One of them is EEG fMRI and one of them is EEG TMS. So if you want to combine EEG with fMRI measurements, then you really have to make sure the equipment is safe in the MR environment, that it works in the MR environment, that it is non-magnetic, and that the data is usable. So for the electrodes, you need to work with passive electrodes. This does not work with our active electrodes here. So that would be the brain cap or also the sponge-based RNet MR that can be brought into the scanner. And we also have a kind of recent-ish development. These are these carbon wire loops. These are things you can add to your EEG cap, to your electrodes, to record motion artifacts inside the scanner that are otherwise extremely hard to clean from your EEG data. So this is the electrodes you would go for. And then for the amplifier, you really just have one choice with us. That is the brain amp MR or the brain amp MR Plus, depending on if you need DC recordings or not. And yeah, this amplifier is really made for the MR environment. It has a very high sampling rate, high filter cutoffs, a large recording range, and this is made to really depict the fMRI signal, the fMRI artifacts, as well as possible, so you can remove them properly from the EEG data in retrospect, and then have nice EEG recordings despite working in this difficult environment. And then the other thing I mentioned is EEG with TMS. So here I'm not talking about pre-post EEG measurements, but really recording EEG and stimulating with transmagnetic stimulation at the same time simultaneously. So if you want to do this, you will have some requirements on the electrodes so you get good data. First of all, you need gel-based electrodes with low impedance, and you need to have a flat profile if possible. This is because you want to come as close as possible to the head with your TMS coil to stimulate, and you don't need to increase the stimulation power like crazy, but you can get really close to the scalp. Yeah, the cables should also be flexible in a way that you can route them in all directions. This is because you might stimulate from different directions, and you should always try to get the cables orthogonal to the handle of your TMS stimulator. So this is also possible with our specific active electrode systems here, or with the brain cap TMS that has cables that are not fixed in the fabric, but that you can move around, and that have specifically flat ring electrodes. And then concerning the amplifier, you will need a very high sampling rate, a high filter cutoff, and a large recording range, similar to the fMRI requirements. Here again, you want to depict the TMS artifact as well as possible to avoid that it spreads into the rest of your EEG data and, let's say, messes with your recording more than necessary. Okay. There are other solutions. So having talked about this, one special case I also want to mention is EEG eye tracking. Not because it's very challenging per se, because it doesn't really interfere with each other, but we have a close partnership with Tobii, and we have a solution that is very nice and streamlined for EEG eye tracking. So if you want to perform EEG eye tracking, the one thing you will need to take care of is to share trigger signals, synchronization trigger signals in both data streams, the EEG and the eye tracking data stream, that appear at the same time. And then you can align both data sets, and then you can profit off what you find from your eye tracking analysis and use it for your EEG analysis. For example, you mark an event in your eye tracking, for example, a fixation. You mark that in a time, and then you can import the timing of that fixation to your EEG analysis and, for example, calculate fixation-related potentials. For this to work, you need to set up these shared triggers, and you also need to have a pipeline to align the data streams. And we have exactly that. We have it for both the mobile and the lab version. So our mobile EEG eye tracking works best with LiveAmp and the Tobii glasses. And there's just one simple cable connecting the two of them. And then they will share synchronization markers that allow to align the data afterwards. And for the lab setup, you might be using either this is a Tobii Spectrum, or you use any screen-based eye tracker, and then you connect it via the trigger box to our EEG amplifier, and you can share, again, event markers. If you want to do this, you should definitely also have a look at our analyzer solution. So that's the Add Channels add-on. This will allow you to import eye tracking data and eye tracking events directly into your EEG data and perform eye tracking related analysis inside analyzer. We have 2 webinars on this specifically, so if you want, you can take a closer look at that. Now, we also have specific solutions for other scenarios. What is quite popular is, for example, combining EEG with FNIRS. Here you have to take a specific look at the caps and how they combine, like the flexible FNIRS montages with the EEG electrodes. We have specific systems like caps that are meant for MEG, so you interfere as little as possible. We have sleep EEG caps, so these specifically need drop-down electrodes that can record facial muscle activities, so they are very practical. And we have smaller headsets that are optimal for BCI, for small setups, pilot studies that you can take a look at. So basically, to sum up, I want to let you know that you have options. So you can use a lot of different electrodes and amplifiers. Depending on your research question, you can try and optimize your selection, what will best fit your research goal and what will give you the best results, so you can publish your data and be happy, hopefully. I hope this was a bit helpful and didn't confuse you even more about all the different options we have. I have compiled a few tips for you that you can read through, different articles and webinars if you want. And yeah, I hope you can also join us in GatherTown later on, ask all your questions. You can also address questions to sales@brainproducts.com if you want to ask about specific solutions, specific hardware. And yeah, with this, I'm a bit early with my talk. I'm sorry, Shiva, but that gives us maybe a bit more time for questions.
Thank you, Laura. That was very nice. So I have a couple of questions that are related from Subhashini, and she is interested in the sports EEG acquisition during sports. And could you say more? Her question is it really possible to acquire EEG during movement, and won't the signal and data points be affected? For example, if they're doing bungee jumping or something.
Okay, so for the bungee jumping study, I assume the program, the time they are most interested in is probably the second or the 2 seconds before the jump is initiated, right? So this would be the EEG activity pre-jump, but I can imagine that possibly you can also look at what happens in the brain during this dive. So the question is referring to, for example, data loss or data quality. I'm not 100% sure.
Essentially, could you say more about the sports EEG? Like, how is EEG acquired during movement?
Exactly. So movement is definitely going to affect the EEG. Also, if only motor activity will have a huge part in the brain activity, you will see that is maybe not necessarily a cognitive function you are interested in. So you will have to be prepared for this showing up. So if you, for example, if you walk, you will have a clear gait signal in your EEG data. You cannot avoid this. You just have to be you have to know what you're looking at, and you have to have a very clear question, which time point is interesting for you, and then maybe look at several trials to see the clear signal you want. We have sensors that can help with sport EEG. It's very challenging, definitely. So you could, for example, add a force sensor, and that can be mounted underneath the heel, and then you can record the heel stroke clearly. So this will already give you an indication which part of the signal relates to the gait motion or the running motion, and then you can try and ignore this part of the signal and look at the actual brain activity. Or you might want to look at another common application, I think, is, for example, for throwing movement, for basketball or for the archery that I've shown, that you would be interested in the very moment where an action is initiated. And then you could compare experts, sport experts, and novices who are doing the same kind of motion, the same kind of sport movement, and then compare them. And also there, it's crucial that you don't only have the EEG data, but for example, you add sensor data. You add a light sensor, you add acceleration sensors, and you record all the signal that is possible, and then really burn down what in the EEG is referring to what you're interested in. Hope that helps.
Yes, yeah.
And
It's a complex question, of
yeah,
course.
and again, for making sure that you don't lose any data, so you don't lose samples, you can record the EEG data inside the live amp. So you can store it on an SD card. You are not bound to the wireless connection via Bluetooth. You just start the recording and then it will be written safely on an SD card and you won't have any data loss.
Okay. So we have another question here from Anya, and she is wondering, is there any data available on the comfort level with the CGX system?
Hard data, I'm not sure. You will probably hear different things. I personally can wear it for about an hour and then it becomes uncomfortable, but I think it differs quite subjectively. So some people are more sensitive to pressure on the scalp and then they will find it uncomfortable already maybe after 45 minutes or something. So there are some individual differences, but it really does make a huge, it is a huge improvement compared to single pin electrodes because you always have this small grid that presses against your head. And this is very significant. But I would say up to an hour is probably okay.
Okay. And let's see. So then there's a question here. Can I combine brain products electrodes with an amplifier from a different company?
Well, that's a good question. So it depends on the electrode. So our ActiCap electrodes are really meant for our ActiChem Plus or our live amp. You can combine them also with the brain amp that is traditionally for passive electrodes via control box because they need to get powered. And if you can somehow manage to connect this control box to your amplifier, I suppose you could record them. For passive electrodes, it's a bit easier. So some of our passive electrode systems have touch-proof connectors, and they can connect also to the standard touch-proof connectors of your amplifier potentially. So it's not impossible.
Okay, that's and just to go in a slightly different direction involving different interactions between different products, and can you combine different amplifiers to do hyperscanning, for example, in this application? Maybe you could say a little bit about hyperscanning in case people don't immediately know it as well.
Yeah, so for hyperscanning, the goal is to measure brain activity from two different people to see how they maybe align or maybe interact with each other, one following the other in a way. For example, when they're making music, when they're dancing, when they're watching each other in social interactions. And for hyperscanning, the optimal setup is really having the same recording conditions for both participants, because otherwise you might not find these alignments in the data. Yeah, so I would not recommend using different amplifiers. That is the first thing. I mentioned that the brain amp is basically the gold standard because it really records everything on the same clock. But you can also combine data sets a bit like in EEG eye tracking, that you share synchronization markers and then you align them in retrospect. And if the markers are very precise and they appear in precisely at the same time point in both data sets, and there's no drift between the amplifiers, then you can align them nicely afterwards. So you can use all our amplifiers, but I would not mix them. Maybe if you're interested in hyperscanning, a nice thing is to look at the merge channel solution from Analyzer. I don't know if my colleagues are going to talk about it. This is a solution we came up with that will help you merging data sets from different recordings. So for example, from recording two people in hyperscanning, and it will align the data sets automatically and will tell you also if there's an offset, if there's a jitter. So you can look at also the quality of your hyperscanning recordings.
Okay. Right. Here's a question also about a very different kind of application. that essentially, if you want to record auditory brainstem potentials, which amplifier and electrodes would you use, or how would you go about it? Yeah.
So auditory brainstem potentials, I believe, are very small effects and at a very, very early time after stimulation. So you have to have a high timing precision. You have to have very low impedance values, I assume, to have as little noise as possible so it doesn't cover up your effect. And we have a special solution for this. It's the EP preamp. So this is made to really, look at these very early, very small potentials. So if you want to apply that, maybe drop an email to sales@brainproducts.com or to your local distributor to clarify. But this EP preamp works with the ActiGen Plus. Can be combined.
Okay. And maybe we'll take one more question, and then we will head to the next talk. It's again a question about hyperscanning. So suppose you're measuring data from two people. Do you need typically two monitors to monitor the EEG data from two different people?
Yes, so basically it depends on the hyperscanning method, right? So if you use BrainAmp, that will already combine all the data to one data stream from both participants. That will end up on one recording computer and one monitor. If you use the technique where you align the data in retrospect, so you record them at the same time, share synchronization markers, align them later on, then you will need two different recording computers during the experiment. And a third option is to use an independent recording platform, lab streaming layer. So if you can manage to stream your EEG data to the Wi-Fi and then grab it with lab streaming layer, you are recording it with the same recording program, right? So this will again bring the data streams on the same computer clock of the recording program. So this is basically another way to make sure it's aligned. This is possible with all of our amplifiers. So they have connector apps that can stream data to lab streaming layer, and then you would record it with lab recorder. That would then align them on the same clock. And in this case, the recording with lab streaming layer would be on one computer. That would also be one monitor on.
Okay. Yeah. Thank you very much, Laura. We are a little running a bit early, but if it's okay, I think we can transition to the next talk. And this will be about what software do I need for recording and analyzing my data. And this will be presented by Lena and by Michal. And let's see if we can get going. Thank you, Laura.
Thank you.
And Lena will be first. Yeah. And she will be telling you a little bit about a recorder and how to set up your workspace to acquire data and setting up your software. And this will be followed by Michal, who will talk about analysis.
Okay, so hi everyone and welcome to this session about our software. So as Shiva just said, the first part of the session will be about BrainVision Recorder, our software for recording the EEG data. And the second part will be by Michael, who will walk you through Analyzer. So when Recorder was developed like 25 years ago, the basic idea was to create a software that could, of course, reliably record your EEG data. But it was also the aim to create a software that goes with the diverse requirements of neuroscientific research, and a software that should also be able to be used flexibly for different setups. So BrainVision recorder is able to be used with all of our amplifiers, with all of our caps and a variety of sensors. It also allows you to do multimodal recordings, like physiology in addition to the EEG, or you can record the video together with the EEG. Or you can also go into the MR and do simultaneous EEG fMRI recordings. It also allows you to customize for different signals, for different applications and setups, which is shown here and which I will walk you through later on. It also allows you to stream data to other software using our remote control function of the BrainVision recorder. Sorry, it's not going to the next slide. I'm sorry, seems like my PowerPoint just crashed. So now, if we talk about recorder, I think we should talk about the workspace concept. So the idea of recorder is that you have, or of the workspace is, that you can save all your relevant parameters into one file. And this allows you to have easy access to frequently used recording parameters, like the amount of channels you're recording from or your sampling rate. And this also ensures the reliability of the data acquired within the same study. So imagine you're running a lab, and within the lab you have different kinds of studies. Maybe each of the study is using the same amplifiers, but there might be still some setups which are different between the studies, like your sampling rate or the channel numbers. So in the beginning of your study, you could set up your workspace, and in this workspace, you can specify these parameters. And then when switching between the studies, you can always upload the correct workspace. And with this, you will have all the settings as you will need it for this study. So the core functions of the workspace are the data file settings. I will walk you through this in a minute. The amplifier, cap, and sensor specific settings. You can also specify some software filters. And there are also settings for the co-registration of your event series or triggers. So when you create a new workspace for your recorder, the first interface will ask you where to save your data. So by default, it will be on your hard disk under Vision and RAW files, but you can specify whatever folder you like to use. We recommend that you save it on your hard disk to avoid any data transmission problems. You can also specify whether you want to name the files individually or if you want to use automatic file name generation. If you enable this function, there is a prefix you can give to your file name, so each of the names or each of the data recorded within a study will have the same prefix. Added to this prefix will be a number. The number will be counting up for each of your recordings, so you can avoid to overwrite your data by mistake. The next window allows you to set some amplifier and cap specific settings. So here, for example, we see we have ActiveChamp Plus connected. The ActiveChamp Plus comes with a maximum of 160 EEG channels and 8 auxiliary channels. You can also specify, or you have to specify, whether you're using active tri-electrodes or passive electrodes. Then you have a drop-down menu which allows you to select the sampling rate you want to use for your study. Which sampling rate depends on the amplifier, as Laura just explained. Then we have 160 EEG channels available, but maybe we don't want to use all of them. And we can specify here which or how many of the EEG channels we really want to use. Special for the ActiveChamp, we also have to specify which of our EEG channels is the reference channel. And down here we have the cap specific settings. We see here the type of signal we are recording from. So these are EEG channels, and here is in this column the names. In the beginning, recorder has no idea what the name of your channels are, so there will be simple numbers. And then you can either type in the correct name, or you can use this button here and upload a file in which for each of your channel the correct name and the correct coordinates are written in. If you use the standard names of the 1020 system, recorder will know the correct coordinates for each of your channel and will assign it. So as I show you later, in the impedance mode, the correct coordinates can be used. If we want to use the electrode position file, we have to click on this button. Then an interface will pop up asking you whether you really want to use the electrode position file. If you check this button, this box, then you will go to your folder where the electrode position file is located. The electrode position file has the ending BVF and is your BrainVision electrode position file. Usually this file comes with your cap, but if you lost the file or if the cap was handed over to you and you have no idea where the file is, you can go to our homepage and download it there. And if you still can't find it, you can contact us and we can provide you with the file. So if we then import the channel table, you will see that instead of the numbers, you have now all the correct electrode names in your channel settings. And as I said before, if you use the standard names, then recorder will know the coordinates and the names will be always saved in your header file, which allows analyzer later on to use it. So now we go back to our channel settings from the workspace and we scroll down. We see that in addition to the EEG, depending on the amplifier and your settings, there will be bipolar electrodes available as well, and auxiliaries. In the auxiliaries, you can connect your sensors, so you will have the sensors, connect them to the auxiliary board of your amplifiers, and most of the sensors using a different unit than the EEG. So you can check here, say, yes, I'm using a different unit, type in the unit and the gradient and offset if needed, so that the scaling and everything is correct during the monitoring. The next section in the workspace allows you to specify whether you want to use filters and which kind of filters. So we have the raw data saving filters, and we have two kinds of display filters, like the display filter and the segmentation filter. The raw data saving filters will have an effect on your recorded data because they are really applied to your raw data. So if you enable this function, your raw data will be altered. So even though we offer this possibility, we recommend that you don't use it. Instead, you should use the display filters. The display filter will be applied only to the display on your screen, but the recorded data will be unchanged. In the display filters, you have the option to specify a low cutoff filter, high cutoff filter, or a notch filter. You can enable it for all of your channels or for individual channels. And the same filters can also be enabled for your online segmented data. And online segmentation, that's what the next window in Recorder is about. So you can choose whether you want to have an online segmentation or averaging of your data in together with the continuous EEG recordings. If you want to use the online segmentation or averaging, there's a number of functions available. You can choose whether you want to save the segmented or average data together with your continuous EEG data. You can specify different groups of segments or averaging. You can enable which of your markers should be used for segmentation. Then you have, of course, to define the time interval for the segments. You can choose whether you want to use a baseline correction or not. If you want to use an averaging, then you can also specify for how many of your segments the averaging should be done. And last but not least, we should at least shortly have a look at the co-registration of your wind series. So to allow recorder to translate the electrical signals, which are sent from the stimulus PC via the digital port of your amplifiers into markers in recorder, you need to set the digital port settings correctly. Details on this topic will be tomorrow, so this is just shortly so you have heard of it. As a short illustration of the co-registration of your triggers, so basically in most of your experiments you will have a stimulation PC, and on the stimulation PC some kind of experimental control software is running. The stimulation PC is sending a trigger, which is an electrical signal, at each stimulus presentation. The signal is sent via the trigger cable to the digital port of your amplifier. The amplifier is simultaneously recording the EEG data and the data from your sensors, and both is sent to your recording PC on which recorder is running. And the interpretation of the triggers are then depending on your digital port settings, so you really need to pay attention when setting these up. Now, when everything is set up in your workspace, you can save it under a meaningful name. And if you have different studies, there will be different workspaces for each of your study. So whenever you start the recording for a study, you will upload the correct and the corresponding workspace. And with this, recorder will know all the settings you have made during the workspace setup. Now imagine that everything is set up, you have prepared your workspace, you have connected your hardware correctly. Then the next step would be setting up the cap on the head of your participant and then the impedance measurement. As I already said, if you have made the effort of typing in the correct names or using the electrode position file, then all of your electrodes will be shown on a head model, and the impedance values are shown in a color-coded way. So from the color bar, you know which color represents which impedance value. So when preparing the electrodes, you know immediately which of the electrodes might need improvement. Here, maybe this one still. And which color, so where the threshold is for good and bad impedance, depends on which electrodes you're using, and you can specify it as well. So in the last part of the session, I would like to go through how the monitoring of your data looks in recorder. So if we switch to monitoring using this button here, then we will see our continuous EEG data, and if we have selected to use online segmentation in our workspace, we will see the segmented data as well. We have here our continuous EEG data on the left-hand side. Here are the names of our channels, and here are the continuous EEG data. On the bottom, you see your markers, which are received by recorder. So the red bar indicates the time point when the marker was received. The letter is the first letter of your stimulus or marker type, and the number is the binary code of your marker. You will have for your continuous data and for your segmented data a toolbar. This toolbar allows you to change the time interval, for example, if you want to zoom in or out. You can change the amplitude of your signal, and you can change the amount of channels which are shown on your screen. You can then scroll up and down using this button, and here you can activate or deactivate your baseline. And the same is also true for the segmented data. Here you have all of your channels shown in the panels here. And how this looks like in real data is shown in the following video. So here you see the continuous EEG data as they are recorded. Again, here you have your channel names, and there are your continuous EEG data. On the bottom, you see the markers, and you can see why the data are recorded, how recorder is receiving the markers. So they're kind of replacing. You can also see that the segments are changing, and that is because they are calculated for each of the markers, so they are frequently recalculated. Then you can also zoom in or change the number of channels which are represented, so you can see a bit more detail. And here you can also see the time interval and your pre and post stimulus time. So the next one would be also to have a look at the signal from some sensors. Here I would like you to pay attention to the GSR signal. The GSR is, to those of you who are not familiar with this kind of signal, it's the galvanic skin reaction. And this is a quite slowly changing signal. So if we look at it with the time scale of the EEG, we will see nothing but a flat line. But luckily, Recorder offers us the possibility to adjust the visualization of individual channels in a scientific view. And with this, down here, we can change the time interval, for example, and then we will see that we are indeed receiving a really nice GSR signal. So the flat line in this case was only an issue of the wrong settings. Now, after we are done with our monitoring, we can have a look at our recorded data. And there you will notice that it's not one file, but 3 files your data saved to. So you will have a file ending to .eeg, one ending to .vhdr, and one to .vrk. The .eeg file is a binary data file which contains the time series of your EEG. The VHDR file is your header file. In this file, you will find the general information of your recordings, reflecting the settings in your workspace. So you can see the number of channels you are recorded from. You can see your amplifier specific settings, whether you have used software filters, and if so, which kind of software filters were used. There you will also find the impedance values. So the last values Recorder measured before switching to the monitoring will be saved to your header file. And in the header file, you will also find internal reference to the EEG file and the marker file, so that analyzer knows which files belong together. And for this reason, you also should not simply rename your files, because with this, your internal reference might be not valid anymore. And the last of your files will be the marker file. In the marker file, there's a list of markers with all their data point position, timestamps, the marker type, like here it's stimulus, but could be a response or whatever as well. And there you will also have a reference to your EEG file. So they are linked together. And with this, I would like to come to the end of this part of the session. So if you want to learn more about Recorder, there will be another session tomorrow on this topic, but you can also go to our homepage. There is a user manual for Recorder available, but also for our amplifiers. There are press release articles showing tips and tricks for Recorder, as well for different setups. And Recorder is updated frequently, so it makes sense to check from time to time whether the version you're running on your PC is the most recent one. If not, you should consider to download the most recent version and update your Recorder. And then if you run into any technical problems, you're welcome to contact us from the technical support team using this email address. And with this, I would like to thank you for your attention. And I would like to hand over to Michael for the second part of the session, which will be on analyzer.
Thank you, Lena, for starting us. I'm happy to take over. Let me just share my screen. There we go. Okay, hello everybody also from my side. As Lena said, I want to continue our journey about the software with our analysis software, the BrainVision Analyzer. So I'll just start where Lena ended. You have already learned our native BrainVision core data format consists of a file triplet. The data is stored in the EEG file. It's binary coded, so you need a specific software to open this, like our analyzer. And then we have the header file, including the information on the data structure. And we have the marker file containing all the event codes. And Lena also already mentioned, you should not rename the file simply because there are internal links. Here, I just want to add, if you have this problem, you can just drop us an email because we're having a dedicated renaming tool for this that does this job for you. So just send an email to support@brainproducts.com and we can give you this tool. So this standard format is generated from Recorder. However, you can also create this format via exports from Analyzer, for instance, or other software. And then sometimes these files might be a little bit different because you can usually customize a little bit more what you export. For instance, the header and marker file, they might not be in text format, but they might be in XML format. And Analyzer is designed to be actually quite open. So for many file formats, not just for our own. Of course, it can read our own file format, but there are around 30 other formats from other manufacturers or open formats that can also be read in Analyzer. And I have just put a few here on this page, which are those that are very commonly used, but there is a full list on our website, which you can reach via this link, or if you use that QR code. So the question is then, how do you get the data into Analyzer? And the good news is, no matter if you're using our format or if you are using another format, the way is always the same. You do not just import the data directly, but you rather create a workspace. What is a workspace? You simply define paths pointing to 3 dedicated folders. One folder contains your raw data. So in our case, that's the file triplet EEG, VMRK, VHDR files, or any other raw files. Then you have a history folder and an export folder. And they are empty in the beginning when you start your analysis. This is a general folder structure that Analyzer expects. You can, of course, also use the same folder for all 3 types of files. However, we recommend not to do that and rather use 3 dedicated folders so that it's tidier and easier to keep an overview of the files that you have. You can name those folders in any way. We just suggest these very obvious ways of renaming them so that it's clear what's inside. Once you have defined the path to each of these file folders, they are stored in a workspace file. And then Analyzer will scan these folders, and for each raw file that it finds, it will create a history file in the dedicated folder. And then the datasets will appear in Analyzer. And importantly, the raw files will never be altered by Analyzer. And you're also never asked to save your processing and store copies of these files on hard disk, because Analyzer will always on the spot automatically save everything that you do in the history file while you are doing it. So that's why you need this folder, and everything is stored at on the spot. Okay, For the rest of the session, I actually want to show you a little bit Analyzer. So I'm going to switch my view to Analyzer. There we go. This might be a little bit small on your side because there are a lot of buttons in here. So what you can do is using the zoom function that you were shown in the very beginning of our session today. So just click on this plus sign and then you can navigate around and zoom in where you want. So this is when how Analyzer looks when you open it, and I want to guide you once through the interface a little bit. I want to start with this first file here. I'm clicking on the plus sign, and then you see there is a raw data file here. Because if I right-click here and go on the recording infos, if you have recorded your data with our data format, so it's coming from BrainVision Recorder, then you can actually look here into the information that Lena has mentioned before. So this is all the workspace information from Recorder, including the impedance values, and this is still accessible here. Okay. But now let's do like a proper tour of the interface, and where I started is the first important area here. It's the so-called File Explorer, the History File Explorer, and it has two tabs, and we're looking here at the primary tab at the moment, and there are 3 files, and these are 3 single subject recordings, so individual recordings here. In the secondary tab, there's actually nothing right now because this is for group data sets. So once you get through the single recording analysis, then you can create group data analysis sets. Then for the next part, let's actually open some data. I'm using this data set here, clicking on the plus, and now I can visualize this data by double clicking. It will appear here in the middle where we have at the moment our online info, where you can see again the information about important updates and workshops. I'm closing this, and then I double click here to open data. So what do we have here? We have the classical or the standard view, which is all the channels on top of each other. You see here the names in the front, and then the EEG traces towards the right. You can simply click on a name to highlight the channel or multiple, and then if you double click, you can zoom into those channels. If you double click again on a channel label, you just go back to all of them. In order to navigate your data, you have to use the navigation bar down here. You can see that the navigation bar consists of this bar here, which has a lot of red bars in there. So these are actually the markers in your recording. So if I now use these arrow buttons, I can navigate through the data. And when I'm like here with this slider below one of these red bars, you can see this is the corresponding marker here. You can also use these other 2 arrow buttons, which are for smaller steps of 1 second forward and backward. You can also just jump somewhere by clicking in this bar. And for those of you who like to use the keyboard, if you press the control button and then use the arrow keys to the left and the right, you can also navigate the data easily. Together with this navigation bar, you should use the toolbar up here, because this has all of the other important visualization options. For instance, you can use those magnifying glasses here to either zoom in or zoom out and show a different time range. Then the same we have here with scale up and scale down. And you can also show less or more channels. And then if you want to, you can always click these buttons here with the R for reset interval and reset amplitude and go back to the defaults. Again, if you like to use your keyboard, you can press the control button and use the arrow up and down keys for amplitude scaling and plus minus keys for time range adaptation. The next important part of the interface is down here to the right. It's maybe rather small, so make sure to zoom in. This is the status bar. This shows you a lot of different information. And actually, if you right-click here, you can also customize what is shown. And importantly, if you go into the data with a mouse and there is this small crosshair, the status bar will show you exactly on what channel you are with this crosshair, what time point you are at and what data point, and what is the corresponding amplitude value. So you can use this to read out some values on the spot. The next part of the interface is to the right. That's the view toolbox. I just want to say here you can in the end use this if you're creating graphics to add some additional customizations to graphics. So this is not part of analysis functionalities. However, very important otherwise for the whole analysis is what you have on top here, which is the menu. And I want to start from the beginning, which is the file menu. And then I will just walk you now through all of these tabs so that you know where is what. So at first you have the file tab here, and this has a few important options like everything around a workspace. So that means to import data. You have graphics options here and also all of the preference and view settings. Then we have the display tab. This has mostly all options around creating and switching montages. And those are not what you might know as a montage in the sense of referencing, re-referencing the data, but this is rather just meant as a visualization option in Analyzer. Then we have transformations. That's the most important part of the Analyzer interface here because this has all of the main functionalities for data analysis. And this is actually grouped based on the type of function or algorithms. So let me just mention a few that will also become important in the next 2 days of this workshop. For instance, in this dataset pre-processing group, we have the new reference, which is actually the transformation you need for re-referencing the data. Then in this artifact rejection reduction group, we have here this filtering drop-down, and here you have IIR Butterworth filters. We also have here raw data inspection for marking artifacts. And then, for instance, here in segment analysis functions, we have segmentation, which is the tool that you need to cut trials or epochs out of your continuous EEG data. You might see directly some of these transformations, they are grayed out, and actually I can't click them. So if I click here, nothing happens. But this is actually on purpose because Analyzer says, oh, well, it doesn't make sense to calculate an average now because the data is not in segments yet. So it will just not offer you this option because the data that you are looking at the moment is not fulfilling the requirements. One last transformation I want to highlight here is on the very right. It's the MATLAB transformation. So if you ever use Analyzer and you want to add any additional functionality that is not available in Analyzer, you can transfer your data to MATLAB and apply some either custom scripts there or use any available MATLAB toolboxes. Now let me actually skip one tab and jump to exports. As the name suggests, everything you want to export is in here, or most of the exports are in here actually. And you can, for instance, export generic data or markers. You can also get area and peak information, so mean amplitudes and peak amplitudes, or specific file exports like to EDF or BESA format. And then we have 3 tabs here, the add-ins and the macros and the solutions. They are all containing some additional programs that are not in the transformations. That is mostly due to how they have been created, how they are coded. and they can be used on top of the transformations. So for instance, in the add-ins, we have something here that is called troubleshooting. That's a diagnostic tool mostly, but here's also something that's called Analyzer Video, which you can use if you are doing simultaneous EEG and video recordings. In macros, there's nothing right here, but what you can do is actually open here a window to code yourself. If you ever get into the situation of doing this, Analyzer is having an interface for a Visual Basic derivative called Sachs Basic. However, most of the time you will not have to do this, because if you ever need an additional function, it's highly likely that there is already a solution for it, which is a type of macro. However, the solutions have been written by Brain Products support, and you can actually download them for free from our website. So they do not come with the Analyzer installer. You have to install them separately, but you can just download the installer from the Analyzer downloads page. And in here are also, like in the transformations, different groups. So for instance, also here you have some additional exports. The next tab is History Template, and this is quite useful because this is our way of automating analyses. So you do not have to write any code in Analyzer to do automation. You can simply create a template from your processing pipeline and run that across other data sets. So this is highly convenient, actually. And lastly, we have the Help tab. Well, there are a few helpful things in there. For instance, the user manual. And then we have also this How to Cite Us tool here, where you can, for instance, grab the citation and take it to your manuscript. And there are also some support tools in here. Next, I want to talk about views, now that you know the interface. So we have here by default this standard view that I mentioned earlier, but you can always also look at the data in a different way. So if you right-click in the interface here and say switch view, let's maybe use the head view. Now we're having exactly the same data here, so 10 seconds of data that we looked at before, but now the channels are not anymore above each other. They are in these small boxes, and the box is in the position on a map of the scalp where this electrode sits. So this can be a nice way of showing specific things. Of course, not always useful, but for specific situations, this might be a nice graphic that you can also customize. If I go back, switch view to standard, I'm back where I started. However, there's a different type of view, which is a so-called transient view. If I mark some data, maybe here, I can open another view, for instance, an FFT view that is opening in a different window. And this is dynamic. So let me actually make this a little bit larger. Scale the amplitudes up and zoom in. So now I'm getting a frequency spectrum, but only for this marked area. So, and if I move this around, you see also how this frequency spectrum is dynamically adapting. Or also if I would make this bigger, it would also change. So this is a very nice way of inspecting your data or creating some graphics. There's also something here that is like a topography. I will show this later. And the important thing, though, is these are just views. They are temporary. So when I close this by either clicking here on the X or by using the escape button on my keyboard, they disappear. So nothing is stored, but you can use this very nicely for making your own graphics. Okay, let's now look a little bit into how to do some processing in Analyzer as the last part of the session. So for that, actually, first you need to understand how to apply a processing step, and this is rather easy. You always have to make sure that the node, the history node, this is what we call that here in this history explorer, that is data is shown in the view, and then you can just apply something. And the new transformation is always applied to the data that is visualized at the moment. And then analyzer will store the parameters that you apply in the history file, but they actually applied on the fly. So if I navigate through the data, analyzer will always on the fly apply the parameters. This is actually very fast and efficient way of showing the data. And there are some cases, though, where it also makes sense to store a copy of the data on hard disk. And we call this caching in Analyzer, and you will find this option here and there, for instance, in the filters, because it's recommended to cache or to store the data when you are filtering. And also in some special cases where you're doing very resource-intensive processing, this can help speed up the processing. But let's now actually look at the processing tree. And for that, I have prepared one in this additional data set. I'm right-clicking here and say expand all. And now you have here this full history tree. And you can see now that this is built up in a certain way. So it starts from the raw data. And then we have here one point that is always stepwise added. So always on the previous step, we're having the next one and so on. So a linear processing pipeline, so to say. And this is because in the beginning, when we do our pre-processing, we usually apply one step to the next one and to the next one and so on. However, at some point, you see then that this processing tree is branching out. And in this example here, there are actually two things where we branch out. The first thing is we're having here this ERP analysis. You will hear more about this also in the rest of the workshop. Then we have one condition here and another condition here in parallel. And then you see that the nodes that are here, they are actually the same because we do the same processing for each condition. Below, we have then again two sub-branches of this tree, and this is because in the same data set here we have two experiments, an ERP experiment and also a resting state with a frequency analysis. And this has again two conditions. So you can see that in this small processing tree here, I have actually realized two different experimental analyses and two conditions each. And you can see already that it's very obvious here what has happened in every step because of the naming. And also, actually, Analyzer is giving you even more information automatically. If I right-click here on this new reference node, I can, for instance, go to Operation Infos. And then it tells me exactly what I have done there. So Analyzer is recording automatically the parameters of what you have been doing. Also, if you right-click again, you see properties here and markers because Analyzer can at every step tell you what are the current data properties and what are the current markers, because this might change actually in the data from step to step, since you can manipulate both the data properties and the markers. Okay, and now at the end, I want to show you just a few additional visualizations. So let's assume we are having processed all our analysis tree here and arrived at the average of our ERP analysis. I'm double-clicking here. And this is the ERP to the deviance. So this is a P300 example data set where we are looking at an auditory oddball experiment. And you see already there is a different view here. This is the grid view where for this short ERP, which is just 1 second long, you get the trace of the EEG in a box for each channel. And if you again zoom in by double-clicking on a label, let's say here on this centroparietal electrode, CPZ, you see the trace for this specific condition. What you can do now is, for instance, mark some data, like this peak here, going in the positive direction, and then get a mapping view. And then you have a topography very easily, where you see now the mean amplitude in this time window here. If you close that again, You can also make an overlay very easily. For that, I'm just going to the other condition with the standards. I'm clicking and dragging that over here and dropping it in the view. And now you have in black one condition and in red the other one, and you can very nicely see the difference between these two ERPs to two conditions, which you can now, for instance, make a graphic for your manuscript, or you can now base some further analysis steps on this and, for instance, export some data for statistics. And with that, I'm already at the end of my short tour through the analyzer software. And I think we can go to the questions.
Right. Thank you, Michael. And maybe just wait for Lena. Yes. So I have a few questions for you. Since you both gave a very nice overview of how to record and then how to analyze data. So the first question is to Lena. So could you explain a little bit like why generally you don't recommend the use of raw data filters during acquisition?
Well, if you use the raw data saving filters, you will change the data you're recording. So it's not like the real raw data anymore. And the problem is, if you have chosen a wrong filter settings, for example, there's no way of going back. So yeah, you really change your data. And as Michael already showed you, you have the possibility to apply the filters later on during data analysis. But there you can change the filters. So in case something is not as expected, you can change them, but not if you use this as a raw data saving filter.
Okay, makes sense. And now I'll just arrange the questions so we jump back and forth between both of you. So I have a question for Michael. You mentioned about loading data in BrainVision format into MATLAB. Is it also possible to load data into Python-based analysis tools?
Yes, that's also possible. So in that case, we do not have a nice bridge that can directly go into Python tools like it can go into MATLAB. But so our data format is rather common because our amplifiers are widely used, and therefore a lot of open source software and MATLAB tools and Python tools are usually open source. Somebody programmed also a tool that can load our data format. So certainly you can load the data also in Python tools. For instance, the MNE Python is a rather commonly used one that can read our data format. In MATLAB, for instance, EEG Fieldtrip can also do that. The important thing is you can load our standard data format with that. So the one that we looked at before with the 3 files. So that does not have just to be raw data. You can also export this from Analyzer, and this is, you are still looking at my screen. So in export, there's this generic data export. And with that, you can create a file triplet at any point of your processing pipeline. And then you go with that data into another software where you can add additional processing steps based on the processing you have done here.
Okay. And here's another question, this time for Lena. So during recording, suppose you want to only look at a subgroup of channels. Is there some easy way to do this? Is it possible in Recorder to do this?
Yes, it is possible. So you have different options. During recording, you can decrease the number of channels, as I shown you during the presentation. But if you want to see a specific subtype of channels, like some frontal electrodes and some occipital electrodes. You can also right-click on the channel names, and by doing so, selecting some of your channels, and then double-click on the last one you marked, and then you will see only the selected subset of channels. Also, you can define a specific display montage, and but this you can't do during monitoring, so during recording. You have to specify it before, but then you can switch to the montage during your recording. So if you say, I know already that I want to see a specific set of electrodes, and that's always the same for each of my recordings, then I can specify the montage and switch between all of my channels and the specific subset.
Okay. Yeah. Here's another question, this time for, I think best suited for Michael. It's a question from Tatiana, and she's asking, could you say a bit more about how Analyzer does, what visualizations for FFT are available for FFT analyses and visualizations?
Yeah, sure. So basically, what our, I'm just showing this again, maybe not on the, let me go back to some continuous data. So first of all, for an FFT analysis, the classical visualization of the results would be just a spectrum to display. So you can do this with this transient view here, and then you have basically the spectrum right here. So you don't see anything in the beginning, but let's now zoom in. So for instance, in this channel here, and then you have here the option of this spectrum. So this shows you also where a peak is in what frequency band. The frequency band definition can also be customized. So this is in the view preferences, which you find here in the file menu. And yeah, you can see here, of course, this is now on some continuous data. This is not a really smooth spectrum, but if I show you, for instance, in our second pipeline here that I have as an example, there we have actually analyzed an alpha spectrum, so alpha power for a resting state. And if you then, for instance, go here to the average power eyes open, double click here. Now you see the view is pretty much the same. You see it now again for each electrode separately, because this is averaged data, and this is the mean spectrum for each electrode for a short period of that has been averaged over what do we have here, 1 second segments, so continuous 1 second segments. And then you see here the spectrum for this kind of visualization. And then you can also again do an overlay with the other, for instance here, this other condition. And then you see directly here, the other condition is shown as this red line, that there is an alpha peak here because this is eyes closed, while this one is eyes open. So this is like the standard visualization for frequency data in Analyzer. However, we also have other frequency or spectral transformations that is mostly the wavelets transformation, which is a time frequency method. So in that case, the visualizations will look a bit different. You will get a plot where you have frequency on one axis and time on the other one, and then get this typical heat map style visualization.
Okay. And maybe we take one final question before we move to the next. This is one for Lena. So besides like triggers from the hardware, if something unexpected or unplanned happens during the recording, is there a possibility to mark it in my data, just like an event?
Yes, it is. There's a function called the annotations. So you can press the annotations button at any time point. Then there will immediately a marker in your data, but for the beginning it will be only question marks. And then you can type into a window what happened, like a sudden movement or a distraction to your participant, or you ask them to close the eyes, for example. The other possibility is to predefine annotations before you start your recording, and then you can simply press a number on your keyboard, and then the predefined annotations would appear in your recording together with all of your other triggers in the marker file.
Okay. Thank you very much.